start?
Yeah, teacher ግን የባለፈውን ሚስ ያደረግነውን ክላስ ነው የምንማረው ወይስ የዛሬውን ክላስ በስኬጁሉ?
አይ ሚስ ባረግነው ነው የምንቀጥለው ስኬጁሉ እንደዛው ስለሆነ።
ኦኬ ስለዚህ የሚስ ያደረግነውን ዛሬ ነው ማለት ነው አይደል?
የባለፈው እንማራለን።
ኦኬ እሺ
አይ ቲንክ ስካርብ ነው አይደል ስኬጁሉ?
አዎ ስካርብ ነው።
ስካርብ እንጨርሳለን።
እሺ
ባለፈው ሪካርዲንግ አገኘሽው አይደል? ሁለቱም?
አዎ አግኝቼዋለሁ።
ሰርቶልሻል አይደል? ሁለቱም?
አዎ ሰርቶልኛል ነው። ጀምሯል።
Okay. Our today's session is RNA structure and the transcription process. In the previous class, we discussed about the replication process, and that involves the participation of the DNA strand for the synthesis of new DNA strands. And it was a bidirectional synthesis process, as we said. And the next step of the central dogma of molecular biology is the transcription process, in which the new RNA structure is synthesized from the parental DNA molecule. And we call this the whole process transcription. And the major type of RNAs which are synthesized under this process are the messenger RNA, the transfer RNA, the microRNA, and other types of RNAs, which are very important for the final translation process of the nucleotide sequences to the proteins are participating. So, when we see the structure of RNA, the building unit of RNA is the ribonucleotides, as we discussed in the first nucleotides part as well. ልዩነቱ ምንድን ነው? RNA the single stranded ነው አንዱ ልዩነቱ። ሌላኛው ልዩነት ባለፈው እንዳየነው the RNA contains the ribose sugar and the DNA contains deoxyribose sugar. When you see on the screen, the ribose sugar part is there. On the second carbon, it has OH group below the ring. ይሄ ያው ribose sugar ስለሆነ ነው። OH በሃይድሮጅን ከተተካች ሪሙቫል ኦፍ ግሩፕ ካለ ዲኦክሲ ribose sugar ይሆናል። ሌላኛው as common uracil is found in RNA while thymine is found in DNA. ሌላው RNA structure ላይ የምንመለከተው ምንድነው? The nuclear DNA is in a secondary structure as it has a double strand form, but RNA is in the primary structure. From the types of RNAs, only the transfer RNA has a tertiary structure after undergoing the post-transcriptional modification processes. There are various processes. So, the structure of transfer RNA is also called the clover leaf structure of transfer RNA ይባላል። ቼኑ ስትራንዱ እየተጣጠፈ ነው ያንን ስትራክቸር ፎርም የሚያደርገው። ሶ እንደምታዩት ሌላው ስትራክቸሩ ላይ even if it's a single strand, it has 5 prime end phosphate group. እንደገና 3 prime end OH group አለ። ባለፈው እንደተነጋገርነው when the fifth carbon of the sugar unit is participated in the phosphorylation process, can call it it's 5 prime end ነው የምንለው። እንደገና 3 prime OH group ከሆነ ደግሞ በአንደኛው ሳይድ ላይ ለቦንድ ፓርቲሲፔሽን የሚሳተፈው 3 prime end እንለዋለን። RNA ላይም the same thing ነው ያለው። The ribonucleic acids they are present in the nucleus as usual, and they can also be found in the cytoplasm of eukaryotic cells. Of course nucleusም ኖርማል የሆነ ኒኩሊየር ሜምብሬን ያለው eukaryotic cell ውስጥ ነው። ነገር ግን RNA ኒኩለስ ውስጥ ከተሰራ በኋላ post translational modification transcriptional modification ያካሂድና ወደ ሳይቶፕላዝም ውስጥ ይሄዳል። The reason for that matter is the translation is taking place in the cytoplasm, ribosomoች cytoplasm ውስጥ ስላሉ። ነገር ግን ያው ሳይቶፕላዝም ውስጥም ኒኩለስ ውስጥም ይገኛል ማለት ነው, especially in the eukaryotic cells. So they are involved in the transfer and expression of genetic information. And that genetic information is transferred from the promoter region in the DNA to the proteins. ምክንያቱም የተወሰኑ ጂኖች አሉ, genes of proteins, and those genes will be replicated to the new DNA strand. Then from that DNA strand, those genes will be transcribed to the messenger RNA, and other RNAs will be synthesized in that translation process to synthesize the specific protein of that gene. And they act as primers for DNA formation, as we discussed in the replication process. In order to get free 3' end for starting or initiating the DNA synthesis process, the DNA polymerase requires RNA primer to start to get 3' end ነው. ዛሬ ፕራይመሪ በፕራይሜዝ አማካኝነት እንደሚሰራ ባለፈው ክላስ ላይ ተነጋግረን ነበር. So some of the RNAs act as enzymes as well as coenzymes. You can mention the RNAs. የዚህ RNAs መገኘት የኢንዛይምን ዴፊኒሽን ራሱ ቀይሮታል ብለናል. ከዛ በፊት all enzymes are proteins ነበሩ. Due to the identification of RNAs, it's not protein but it has catalytic activity. And the definition of enzymes is converted to all enzymes are proteins except RNAs. The other one, RNA also function as genetic material for viruses, including HIV. Nowadays it's COVID also. It's an RNA virus. When you see the chemical nature of the ribonucleic acids, such like that of the DNAs, RNAs, they are also polynucleotides and unbranched polymeric molecules. And the RNA polymer, there are purines and pyrimidines, of course, and the nucleotides are linked together through phosphodiester linkage. As we said, the RNA has but it's a single strand. However, they differ in a group from DNA in several ways. Most common are RNA is commonly smaller than DNA in size, as it is a single-stranded as well. They contain ribose sugar instead of deoxyribose, we already discussed. And uracil instead of thiamine, it is also a common concept. But unlike DNA, most RNAs exist as single strands that are capable of folding into complex structures. But the most folded complex form of 3-dimensional form of the RNA is found in the transfer RNA, as we said. And there are mainly 3 types of RNAs in all prokaryotic and eukaryotic cells. Those are messenger RNA, transfer RNA, the ribosomal RNA. Those 3 types differ with each other. The first one in size, the second in matter of function, and the third one in terms of stability. When you see the messenger RNA, it accounts for 1 up to 5% of the cellular RNA is the total. And when you see its structure, majority of messenger RNA has a primary structure. It's normal stranded, okay, and they are single-stranded linear molecules, messenger RNAs, and they consist of 1,000 up to 10,000 nucleotide sequences in a single messenger RNA molecule. And messenger RNA molecules have free or phosphorylated 3', and 5'. This can be betterly described on the previous diagrams as we have seen. 5' in this free phosphate indole group, and the 3' in this 3' OH group. And messenger RNA molecules have different lifespan because we said that one of the basis for their difference is the stability, their stability. So they have different lifespan, and their lifespan ranges from the few minutes to the days. Rather, eukaryotic messenger RNA are more stable than prokaryotic messenger RNAs. One of the reasons for this is the eukaryotic messenger RNAs undergone post-transcriptional modifications that we will see in later. For example, poly-A tail addition, 7-guanosine methylene addition, 5' capping, and 3A tailing የሚባሉ አሉ። እነዚህም after messenger RNA synthesized, it undergoes those modification process. And those processes protect the messenger RNA from the attack by the nucleases. ስለዚህ ስታቢሊቲውን ይጨምሩለታል ማለት ነው። ፕሮካሪዮቲክ ላይ ነው፣ ምክንያቱም ፕሮካሪዮቲክ ላይ እንደምናውቀው they do not have modified cellular subcellular organelles. ስለዚህ ይህን እነዚህ ፕሮሰሶች ያካሂዳሉ ፕሮካሪዮቶች። እዛ ነው the eukaryotic messenger RNA has more stable lifespan than prokaryotes. Messenger RNA nucleotide sequence is complementary from which it is synthesized or copied. It's known that the promoter region is there in the DNA. From that promoter region, based on the gene sequences from the promoter region, the template strand of DNA, okay, one of the template strands of the DNA is complementarily used to synthesize the RNA. Rather, some eukaryotic messenger RNA molecules are capped at the 5' end. I told you this one. And the cap is methylated guanosine triphosphate, and 5' end capping. There might be various capping processes. However, the most popular one and the most common one in the cells is methylated guanosine triphosphate addition to the 5' end of the messenger RNA. Some messenger RNA contain internal methylated nucleotides. That means the methylation might be occurred in between the nucleotide sequences. In the capping, as I told you, protects messenger RNA from the nuclease attack. Rather than as the 3' end of most of the eukaryotic messenger RNAs, the post-transcriptional process involves addition of polyadenosylate or adenylate tails to the 3' end. Those tails, as I told you, their major function is such like that of the 5' end capping. Their major function is to protect messenger RNA from the nuclease attack. So in prokaryotes, the 5' end of messenger RNA contains a sequence rich in adenine and guanine nucleotides. This sequence is called the Shine-Dalgarno sequence. This sequence helps for the attachment of messenger RNA with the ribosome during protein synthesis, because ribosome has a larger subunit and a smaller subunit. The smaller subunit of the ribosome, the messenger RNA should create a matching ወይም match የሚያደርጉ nucleotide sequences ስላሉት complementary sequence የምንላቸው እነዛን ሲኩዌንሶች ስሞለር ራይቦዞም አርኤንኤ ላይ ያሉ ሲኩዌንሶች ጋር ማች የሚያደርገው ሜሴንጀር አርኤንኤው በሻይን-ዳልጋርኖ ሲኩዌንስስ ነው። ሻይን-ዳልጋርኖ ሲኩዌንስ ኤሪያ it is rich in adenine and guanine nucleotides. So the Shine-Dalgarno sequence major function is it helps attachment of messenger RNA with the ribosome during protein synthesis. Some prokaryotic messenger RNA has secondary structure. Intrastrand base pairing among complementary bases allow folding of the linear molecules. እርስ በራሳቸው ለምሳሌ ቅድም እንዳልኳቸው የትራንስፈር አርኤንኤ ብናየው ፎልዲንጉ ሲፈጠር የታጠፈው ቦታ ላይ ያሉ ቤዞች እርስ በራሳቸው ሃይድሮጅን ቦንድን ይፈጥራሉ። ያኛው ለቦንዲንጉ ቤዝ ይሆናቸዋል። As a basis, the hairpin structure might be formed or look like secondary structures might be formed. Hairpin የሚሉት የፀጉር ማስያዣቺ ከፈት ተደርጋ የምትያዝባት የለች ካርቭድ ሼፕ ያላት እና እንደዚህ አይነት ስትራክቸር ነው ወይ ፀጉር ሲያያዝ የሚፈጥረው ቤንዲንግ ስትራክቸር ወይም ፀጉር አለ ይጥፍ አድርጋችሁ ስታያይዙት መሀል ላይ የምትፈጥረው ቤንዲንግ የለች እንደዚህ አይነት structures እንዲፈጠሩ ይሆናል they leads to the secondary structure formation of the RNA. Messenger RNA is direct carrier of genetic information from the nucleus to the cytoplasm. Replication process is taking place in the nucleus. Transcription process is taking place in the nucleus. After the transcription process, the post-transcriptional modifications are there that we will see in the next. After undergoing post-transcriptional modification, the messenger RNA will be transported out from the nucleus into the cytoplasm. Since the ribosome is found in the cytoplasm, as well as the amino acids required for the synthesis of the protein are found in the cytoplasm. Which is that reason we call it that the major function of the messenger RNA is acting as a direct carrier of genetic information from the nucleus to the cytoplasm. The other molecule of messenger RNA contains information required for the formation of one protein molecule in most of the time or in most of the condition. However, there might be genetic information in present. The genetic information might be present in messenger RNA in the form of genetic code. It's unquestionable. The genetic code has 3 nucleotide sequences. For instance, AUG might be a start code. It may translate to methionine amino acid. So sometimes single messenger RNA may contain information for the formation of more than one protein. Usually, messenger RNA means information በአብዛኛው ለአንድ ፕሮቲን ሞለኪውል የሚሰራበትን ኢንፎርሜሽን ነው። ነገር ግን አንዳንድ ጊዜ እንደ ሁኔታዎቹ አንድ ሜሴንጀር አርኤንኤ ላይ ከአንድ በላይ ፕሮቲን የሚሰራባቸው ኢንፎርሜሽኖች ሊሸከምና ሊይዝ ይችላል ማለት ነው። then when you see the transfer RNA accounts for 10 to 15% of the total cell RNA. And when you see the structure, they are the smallest of all the RNAs. And usually they contain 50 up to 100 nucleotides. እንደምዳየን ሜሴንጀር አርኤንኤ ላይ እስከ 10,000 ኒውክሊዮታይዶች ሊይዝ ይችላል ስትራንድ እስከ 50 እስከ 100 and they are single strand molecules. It's known that they might form a tertiary structure ብለናል. The transfer RNA molecules contain many unusual bases now per one transfer RNA molecule. ከ7 እስከ 15 የሚጠጉ unusual bases ሊኖሩ ይችላሉ per one transfer RNA molecule. So unusual nucleotide bases are the methylated adenine, the methylated guanine, methylated cytosine, and the methylated thymine, እንዲሁም dihydrouracil አለ pseudouridine የምንለው ቤዝ አለ isopentenyl adenine እና etc ወይም ተጨማሪ ሆኖ አንዳንድ አሉ። እነዚህ those are the most common types of modified bases or unusual bases found in the transfer RNA እንላቸዋለን። Those unusual bases are important for binding of transfer RNA to the ribosomes and its interaction of transfer RNA with amino acid transfer RNA synthesis. ምንድነው ኋላ ላይ ለፕሮቲን ሲንተሲስ ኮምፕሌክስ አለ ትራንስሌሽን ኮምፕሌክስ የሚባል ያ ኮምፕሌክስ ራይቦዞም ይዟል ሜሴንጀር አርኤንኤ ይዟል ራይቦዞሞችን ስሞለር ሰብ ዩኒት ላርጀር ሰብ ዩኒት ይኖራል ከዛ ትራንስፈር አርኤንኤ ባይንድ ማድረግ አለበት አማይኖ አሲድ ትራንስፈር አርኤንኤ ምክንያቱም እዛ ኮምፕሌክስ ላይ ኋላ የምናየው ላይ ኤ ፓርት ኮምፕሌክሱ ላይ ምን አለው ራይቦዞም ኮምፕሌክሱ ላይ ኤ ፒ እና ኢ የሚባሉ ሳይቶች አሉት ኤ ሳይት አማይኖ አሲድ ትራንስፈር አርኤንኤ ሳይት ነው ፒ ሳይት ፔፕታይድ ፎርሜሽን ሳይት ነው ኢ ሳይት ኤግዚት ሳይት ነው የተሰራው ፕሮቲን ስትራንድ የሚወጣው ስለዚህ አማይኖ አሲድ ትራንስፈር አርኤንኤዎች ማለት ትራንስፈር አርኤንኤ እያንዳንዱን አማይኖ አሲድ 20ውን እየተሸከመ ያመጣል ማለት ከዛ ወደ ፔፕታይድ ሳይት እየሰጣቸው ይላል አማይኖ አሲዶቹ እየተያዙ ፔፕታይድ ሳይት ላይ ቦንድ ፎርም ያደርጋሉ አንድ ፕሮቲን ስትራክቸር ፎርም ያደርጋሉ ከዛ በኋላ ነው የሚወጡት so for the interaction of transfer RNA with the ribosomes as well as with amino acid transfer RNA synthase enzyme those unusual bases have their own function ግንኙነት ለመፍጠር ይጠቅሙታል about half of the nucleotides in the transfer RNA are involved in the intrachain base pairing in order to form its own structure ማለት ነው as a result double helical segments are formed in the transfer RNA that we will see in the structure and what I told you that the clover leaf structure of RNA የሚባለው and further some bases are not involved in the base pairing resulting in loops and arms formation in the transfer RNA clover leaf structure የምንለው arm አለው loop አለው we will see on the diagram and the folding in the primary structure generates secondary structure for the transfer RNA molecule though the transfer RNAs differ in chain length they have some common features with regard to secondary structure what you call it the clover leaf structure so we'll see the secondary structure of transfer RNA that's called the clover leaf structure the clover leaf structure has various ends that we will see the first one is amino acid arm and where amino acid is attached to the C prime end of the adenosine moiety of transfer RNA. You can see on the structure in this slide. As you can see here, it has 3 prime end and 5 prime end. The 3 prime end of the transfer RNA is ended with CCA nucleotide sequences, and adenosine is the last nucleotide to bind the amino acid with it. ምክንያቱም አማይኖ አሲዶቹ ከዚህ ከ3 ፕራይም ኢንዱ ጋር ነው ባይንድ የሚያደርጉት and ይሄ the 3 prime end እንለዋለን ማለት ነው 3 prime OH ይባላል as I told you 3 prime end has an end as CCA nucleotide sequences in the common base sequence as a 3 prime end the other arm what you call is that the thymine pseudouridine pseudouridine ነች ሳይዝ እንትን ያላት እና ሳይቲዲን አርም ይባላል which contains sequence of ribothymidine pseudouridine and cytidine so we can see on the structure here this can square of እናንተ ስታዩት the right side of this the leaf structure of the RNA line I mean pseudouridine እና cytidine sequence him and arm ነው and as I told you from the Greek word psi it indicates the pseudouridine no thymine and pseudouracilouridine they are the two unusual bases found in the arm no thymine በርግጥ it's usual ነው pseudouridine was of one of unusual bases an anticodon arm which can be used to bind with the codons found in the messenger RNA ማለት ምክንያቱም the codons on the messenger RNA the start codons should complementarily matched with the anticodon which is present on the transfer RNA for starting the translation process of protein synthesis so anticodon arm እዚህ ላይ ስለሚገኝ which can be used for recognition of codon on the messenger RNA you can see on the structure an anticodon arm ምንድነው IGC sequence ነው ብዙ ጊዜ የሚይዘው IGC sequence ሊኖረው ይችላል እና አንቲኮዶን አርም ከስር ያለው ነው በዚህኛው ሳይድ ላይ ስታዩት ከስር ባለው ሳይድ the other one the dihydrouridine እንደገና ሃይድሮዩሪዲን እና ዩሪዲን ሬዚዲዩስ አሉ እነዚህ ሌላኛው አርም ነው hydrouridine ያዘው we said that the hydrouridine is one of the uncommon as you can see here on the left side hydrouridine ብዙ የያዘው አርም አለ ይሄ hydrouridine is one of uncommon bases in the RNA sequence ብለናል ስለዚህ DHU አርም ነው የሚባለው hydrouridine አርም ይባላል በብዛት so as it contains much more of the dihydrouridine residues ማለት ነው the 5' end of transfer RNA is phosphorylated and residue is the guanosine you can see here the 5' end እዚህ ጋር ማለት phosphorylated guanosine አለ እኛው ዲያግራም ላይ phosphorylated cytidine ትመስላለች ግን commonly የሚታወቀው በphosphorylated guanosine ነው 5' end ላይ so the 5' end is here 3' end is here the dihydrouridine end በአርም የምንለው በግራ በኩል አለ thiamidine pseudouridine እና cytidine አርም በቀኝ አለ ከስራ አንቲኮዶን አርም አለ ከዛ the 3' end OH group በCCA sequence የሚያበቃው አለ 5' end guanosyl phosphate የምንለው ማለት phosphorylated guanosine then about 75% of transfer RNA molecules have the extra arm the other one it consists of 3 up to 5 base pairs and it's found between the right side of the thiamine pseudouridine and cytidine and anticodon arm ነው ሁለቱ መካከል ነው የሚገኘው ኤክስትራ አርም ይባላል ይሄ ማለት እዚህ ጋር ያያችኋት on the right side እንደ መስቀል እንኳን ነው ክሮስ ብናየው እንኳን እዚህ ጠርዟ ላይ ያለችው dihydroiridine ይዟል methylated cytidine ይዟል በቀይ ያሉት ኤሪያ ያዘ this is an extra arm ይባላል ተጨማሪ አርም ምንለው 75% የሚሆኑት RNA structures ላይ ይኖራል this extra arm has 3 up to 5 base pairs ነው and it is found between the thiamine pseudouridine and cytidine and anticodon arm በሁለቱ መካከል ነው የሚገኘው ማለት ነው እዚህ ጋር ዲያግራሙ ላይ በደንብ እንደሚያሳየው አንኮመን ያልናቸው ኒውክሊዮታይዶች ለምሳሌ ተጨማሪ ሊኖሩት ይችላሉ for thiouridine it might be present some of the RNA molecules inosine also ምክንያቱም እዚህ ጋር ዲያግራሙ ላይ ካያችሁ አሁን IGC ብያቸዋለሁ አንቲኮዶን አርሙ ላይ it might indicate the inosine then 1-methyl guanosine as you can see here on the structure isopentenyl adenosine also can be found the isopentenyl position is present on the 6 nitrogen or it's formed a bond in the 6th position of nitrogen that's why we call it N6 isopentenyl adenosine ribothiamidine also can be one of the uncommon bases in the RNA molecule pseudouridine we have seen it but it has such kind of this structure hydrouridine has also it is also one of the uncommon nucleotide sequences on the RNA structure when you see some modified bases of transfer RNAs and produced in the post-transcriptional reactions. Most of them are formed after the transcription process in order to take the structure of the RNAs. When you see the function of the transfer RNA, it is the carrier of amino acids to the site of protein synthesis. ቅድም እንዳልኳችሁ ራይቦዞማል ኮምፕሌክስ ውስጥ ነው ፕሮቲን ሲንተሲስ የሚሰራው አማይኖ አሲዶቹን ከሳይቶሶል የተሸከመ ያንዳንዱን አማይኖ አሲድ ባይዘው 20 አማይኖ አሲዶች የራሳቸው የሆነ ትራንስፈር አርኤንኤ ያላቸው ለምሳሌ ሜቲዮናይል ትራንስፈር አርኤንኤ ያለ ሜቲዮኒንን ባይንድ ያደረገው ሲስቲናይል ትራንስፈር አርኤንኤ ሲስቲንን ኬሪ አድርጎ ወደ ሲንተሲስ ቦታው የሚወስድ ወይም ወደ ራይቦዞማል ኮምፕሌክስ የሚወስድ so they are major carriers of amino acids from the cytosol to the in the cytosol itself to the site of its synthesis that means in the ribosomal complex and there is at least one transfer RNA molecule to each of 20 amino acids required for protein synthesis. I told you this one. Eukaryotic transfer RNAs are less stable, whereas prokaryotic RNAs are more stable. I told you the reason for this one. The eukaryotic RNAs undergone post-transcriptional modifications, which can make them be able to resist the attack of the nucleases for a long period of time. ለዛ ነው ሬዚስታንሳቸው ቆይታቸው ከፍ ያለ የሚሆነው። When we see the ribosomal RNA, which accounts for 80% of total cellular RNA, and it's present in the ribosomes, as I told you, and it's common one, by the way. And in ribosomes, ribosomal RNA is found in combination with proteins, and those proteins have their own Speedberg unit or S unit. It's known as ribonucleoproteins, the proteins. And the length of ribosomal RNA ranges from 100 to 600 nucleotides. Smaller than messenger RNA and larger than the transfer RNA. Because transfer RNA the most smaller one. So both prokaryotic and eukaryotic ribosomes contain the ribosomal RNA molecules, as it is a must for conducting the protein synthesis process. Ribosomal RNAs, they differ in sedimentation coefficients. Speedberg unit of sedimentation coefficient የምንላቸው ብዙ ጊዜ ribosomal unitoችን ለመለያየት ይረዱናል. So there are 4 types of ribosomal RNAs in the eukaryotes based on their sedimentation coefficients. They are the 5, 5.8, 18, and 28 Speedberg coefficient, or S Speedberg coefficient, ribosomal RNA molecules. So the 5S ribosomal RNA has this function. 5.8S has this function. So it's based on their sedimentation coefficients. Prokaryotes they contain 3 types of ribosomal molecules, and it has somehow difference with the eukaryotic ribosomal RNA subunits. They are the 5S subunit, or 5S ribosomal RNAs, 16S ribosomal RNAs, 23S ribosomal RNA molecules. And each of those molecules have their own function during the protein synthesis process. When you see the structure of ribosomal RNA molecules, they have secondary structure and intra-strand base pairing between complementary base generates a double helical segment of the loops, and they are known as the domains, the larger domain and the smaller domain. For instance, 16S ribosomal RNA with 1,500 nucleotides has 4 major domains. The 3-dimensional tertiary structure of ribosomal RNA is highly complex structure that we will see now. The function, when we see that the ribosomal RNAs, they are required for the formation of ribosomes. Ribosomes, they contain the proteins, ribosomal RNA subunits as well. The 16S ribosomal RNA, 16 sedimentation coefficient unit RNA is involved in the initiation of protein synthesis process, because each of the እያንዳንዱ S unit ያላቸው ribosomal RNAዎች have their own function. ስለዚህ 16S subunit ካለን in the prokaryotes ማለት ነው 16S ribosomal RNA involved in the initiation process of the protein synthesis, and initiation complex ይባላል, translation initiation complex ይባላል። ምክንያቱም as we will see now, each of the processes, transcription and translation, they do have initiation, elongation, and termination. So the initiation process የሚያካትተው the formation of the translation complex የሚባሉት የነሱን መሰብሰብ ነው። ራይቦዞሙን large and smaller subunit, transfer RNAዎቹ with their amino acids, the enzymes, እነዚህ ሁሉ ሌላ other factor proteins to initiate the process እነዚህ መሰብሰብ ነው። So when we see the diagram on the left side there are prokaryotic ribosomes, and on the right side is eukaryotic ribosome. The prokaryotic ribosomes, as you can see, they have smaller subunits and larger subunit. Both together, we can call them the 70S subunit. ነገር ግን when they become separated የተለያየ ዩኒት ነው የሚኖራል። ሴግመንቴሽን ኮኤፊሽንት ዩኒታቸው ይለያያል። ራይቦዞማል ኮምፖዚሽናቸው ነው እያየን ያየነው። 50S እና ፕሮካሪዮቱ 30S ይባላል ሰፓሬትድ ሲሆን። ስለዚህ ስሞለር ሰብ ዩኒት ለብቻው ከሆነ 30S ነው ራይቦዞማል ዩኒቱ የያዘው አጠቃላይ። ላርጀር ሰብ ዩኒት 50S ነው። So on the larger subunit contains 5S RNA, 23S RNA. Totally 50S. Rather than that, it contains around 31 proteins. The 30S subunit ደግሞ 60S RNA የያዘው majorly, then 21 protein units and prokaryotes ላይ ሲሆን. However, when you come to the eukaryotic ribosome, the total complex is 80S ribosome unit ነው ያለው. The larger subunit is 60S. And the 60S subunit contains 5S RNA, 20S RNA, and around 50 proteins it contains. Rather than it has additional 5.8 S RNA. The smaller subunit of eukaryotic ribosome contains the 40S. It's 40S unit member, smaller ribosomal RNA. Segmentation rate totally ማለት ነው 40S ነው። ከዛ it contains 80S RNA ይዟል around 30 proteins. So the number of proteins in the eukaryotic ribosomal subunits varies somewhat from species to species, especially eukaryotic. The large ribosomal subunit catalyzes formation of the peptide bonds that link amino acid residues in the protein. ምንድነው እንዳልኳችሁ ኤ ሳይት ቅድም ቀጥሎ የምናየው ትራንስክሪፕሽን ላይ ፒ ሳይት ፒ ሳይት አለ። ፒ ሳይት ላይ ፔፕታይድ ቦንዶች ፎርም እየሆኑ ነው የሚሄዱት። እያንዳንዱ አማይኖ አሲድ ትራንስፈር አርኤንኤ የራሱን አማይኖ አሲድ እያመጣ ኤ ሳይት አማይኖ አሲድ ትራንስፈር አርኤንኤ ሳይት ላይ እየገባ ያንን አማይኖ አሲድ ለማን እየሰጠ ነው የሚሄደው? ለፔፕታይድ ሳይት ላይ ለሚያድገው ለፔፕታይድ ቼን እያመጡትን አማይኖ አሲዶች እየሰጡ ይሄዳል። ያ ፔፕታይድ ቼን እድገቱ ግሮዙ እስከ ካታላይሲስ information of that peptide bond is taking place on the larger subunit layer. The smaller subunit, it binds the messenger RNA and it also acts as a binding place for the messenger, the codon on the messenger RNA and anticodon on the transfer RNA, and responsible for the accuracy of translation also. By ensuring correct base pairing between the codon in the messenger RNA and anticodon on the transfer RNA. So those all are the functions, majorly the 3 ones are functions of small ribosomal RNA subunits. So we'll see now the transcription process, and we call it the synthesis of RNA by using the DNA as a strand. When we try to give a little clue regarding this in comparison with the DNA replication process, as you can see here, polymerase enzyme they synthesize DNA and RNA. No? DNA polymerase enzyme is RNA polymerase enzyme also. When you see DNA polymerase, the template strand is a DNA. It requires also RNA primer, we said, in order to start the new DNA synthesis. Okay. Rather than that, the DNA polymerase requires deoxyribonucleotide triphosphates as substrate, and it's synthesized in 5' to 3' direction. Now, primary ይፈልጋል ብለናል. When you come to RNA polymerase, it uses DNA strand as a template, yes. However, it uses only one of the DNA strand for a template, as RNA is a single-stranded molecule. And the primer is not required for the RNA polymerase enzyme to start the transcription process. The other one the mispaired deoxyribonucleotides in the during synthesis of DNA can be removed by the 3' to 5' exonuclease activity of the DNA polymerase, especially one. Okay. And it has a proofreading activity also, we said. But when it comes to the transcription process, the mispaired nucleotides cannot be removed because RNA polymerase does not have 3' to 5' exonuclease activity yellow. 3' to 5' direction ላይ ፓረንታል ዲኤንኤ ስትራንድ እንበል እዛ ውስጥ ያሉትን የተሳሳቱ ኒውክሊዮታይድ ሲኩዌንሶችን ቆርጦ ማውጣት አክቲቪቲ የለውም አርኤንኤ ፖሊመሬዝ አይችልም። ዲኤንኤ ፖሊመሬዝ ግን የአክቲቪቲ አለ። DNA polymerase rather than that, it has higher fidelity to synthesize DNA. In our previous class, we said that in most of the condition, the DNA synthesis process has higher accuracy. That's why most of the world people have normal genetical በጣም ሬር ነው DNA synthesis ላይ ወይም DNA polymerase የሚፈጥረው ሚስቴክ። But when it comes to RNA polymerase, the low fidelity RNA synthesis might be taking place. So synthesis ላይ ግን ስናይ both of them are synthesized in 5' 3' direction. Proofreading በርግጥ ለምንድን ያን ያህል ብዙ ማያስፈልገው ፕሮሞተር ሪጅን ላይ ባይንድ ነው የሚሆነው RNA polymerase. DNA polymerase ስናየው ሙሉ ቼይኑ ነው ፓረንታል ስትራንዶቹ ኒውክሊዮታይድ ሲኩዌንሳቸውን እንዳለ ኮምፕሊመንታሊ በሁለቱም ሳይድ ሁለቱም ቼን ሲንተሳይዝ እየሆነ የሚሄደው። however from during RNA synthesis the RNA polymerase is binded with only the promoter region of the selected nucleotide strand of DNA. እና ቴምፕሌት ስትራንድ እና ነን ቴምፕሌት የሚባሉ አሉ። እሱን እናያለን። Now, when you compare the enzymes DNA and RNA polymerases, nucleotide synthesized in both of the condition from 5' to 3' line, DNA polymerase line, RNA polymerase line. However, the nucleotide synthesized with DNA in the DNA polymerase case and RNA in the RNA polymerase case, the required template. That means, in order to copy it from 3' to 5' direction. ቴምፕሌቶቻቸው ለሁለቱም ኢንዛይሞች ዲኤንኤ ነው። required substrates ለዲኤንኤ ፖሊመሬዝ ዲኦክሲራይቦኒክሎታይድ ትራይፎስፌትስ ሲሆኑ ለአርኤንኤ ፖሊመሬዝ ደግሞ ራይቦኒክሎታይድ ትራይፎስፌትስ ናቸው እዚህ ጋር የተጠቀሱት። ለምሳሌ ያው ዲኦክሲ ሆኖ ራሱ ዲኦክሲ ታያሚዲን ትራይፎስፌት ሲሆን እዚህ ጋር ዲኦክሲ ዩሪዲን ትራይፎስፌት ነው። required primer ስናይ RNA polymerase ቅድም እንዳልነው small fragment of RNA ይፈልጋል as a primer. However, DNA polymerase requires a small fragment of RNA. Okay, but the RNA polymerase does not require a primer for starting the synthesis process. Proofreading activity proofreading activity ከምን ጋር ተያይዞ 3' to 5' exonuclease ኮፒ ብቻ ሳይሆን exonuclease activity ላይ ተመጣ DNA polymerase በፊት እንዳልነው ፓረንታል DNA ስትራንዱ ላይ ያሉ አላስፈላጊ የሆነ ፔሪንግ ካለ ያን ኒውክሊዮታይድ ቤዝ አውጥቶ መተካት አቅም አለ because it has a proofreading activity 3' to 5' exonuclease activity ነው። proofreading by the way ኮፒንግ ሊሆን ይችላል exonuclease activity ሊሆን ይችላል። ኮፒንግ ላይ ኒውክሊዮታይድ ስትራንድ ኮፒንግ from 3' to 5' ሁለቱም አላቸው ብለናል እዚህ ቴዎሪ። but proofreading activity ሆኖ 3' to 5' exonuclease activity ካለን DNA polymerase has that activity but RNA polymerase do not have that activity. As I told you before, the RNA synthesis is taking place in the nucleus. Transcription process is taking place in the nucleus. And it undergone post-transcriptional modifications, as we will see. Addition of poly. ብዙ አዲኒን ያላቸው ኒውክሊዮታይድ ያላቸው ጭራ መስራት ነው። ፖሊ ኤ ቴል እኮ ማለት ብዙ አዲኒን ያለው ጭራ ማለት ነው። በ5' ኢንድ ደግሞ ካፒንግ ላይ ይሄ 3' ላይ ነው። ፖሊ ኤ ቴል ኢን ዘ 3' ኢንድ 5' ሚታይሊን ጓኖሲን አዲሽን ኦር ካፒንግ ደግሞ 5 ፕራይም በኩል ነው ያኛው የሚጨመረው። ሁለቱም ካከናወነ በኋላ እዚህ ጋር ዲያግራሙ ላይ እንደምታዩት ሜሴንጀር አርኤንኤ ነው it will leave the nucleus and it will enter into the cytoplasm for undergoing the protein synthesis that is taking place in the complex with the ribosomes and the transfer RNA. እዚህ ጋር ለምሳሌ የምታዩቸው larger ribosomal subunits መጀመሪያ ሲመጣ it comes with the starting amino acid transfer RNA. እዚህ ጋር እንደምታዩት ዲያግራሙ ላይ ብዙ አማይኖ አሲዶች አሉ፣ ብዙ ትራንስፈር RNAs አሉ in order to carry them to the site of the protein synthesis. ከዛ እየተሸከሙ ያመጣሉ ወደዚህ። ኤ ሳይት ያልኳችሁ እዚህ ጋር አለች። ይሄ ፒ ሳይት ነው፣ ፔፕቲዳይል ሳይት ነው። ኢ ሳይት አለ። ኦኬ ሶ እዚህ ሳይት ላይ የመጡ አማይኖ አሲዶቻቸውን እየሰጡ ሲሄዱ ይወጣሉ፣ በኢ ሳይት ይወጣሉ። ማለት እየወጡ ሲመጡ ፔፕታይድ ቼኑ እዚህ ጋር እንደምታዩት እያደገ ነው የሚሄደው። ነገር ግን ሜሴንጀር አርኤንኤ እዚህ ጋር ፖስት ትራንስክሪፕሽናሊ ሞዲፋይድ የሆነ ሜሴንጀር አርኤንኤ ስሞለር ሰብዩኒት ጋር እሱ ኮዶኑና አንቲ ኮዶኑ ትራንስፈር አርኤንኤ ያለው ማች ያደርጋሉ። እንዲያውም ስሞለር ሰብዩኒትን ራይቦዞማል አርኤንኤ ሰብዩኒት እና ላርጀር ራይቦዞማል አርኤንኤ ሰብዩኒት ኮምፕሌክስ ይፈጥራሉ። ኢንዛይም ፓርቲሲፔሽን አለ። ይሄ ሙሉ ነው ትራንስሌሽን ኮምፕሌክስ የሚባለው ኋላ ነው የሚያስፈልገው ዲያግራሙ እንዲገልጽ። So as an introduction part, the synthesis of RNA from DNA template is called transcription. No, already ተነጋገርነው። The genes are transcribed by RNA polymerase enzyme and that generate transcription process generates a single stranded RNA identical in sequence with the exception of uridine in the place of T to one of the strands of the double strand of RNA that's acting as encoding strand. The DNA template is copied the 3' to 5' direction. ከ3 ወደ 5' direction እያነበበ ማለት ነው። ሲንተሳይዝ ሲያደርገው 5' to 3' direction ነው RNA ትራንስክሪፕቶቹ እየተሰሩ የሚሄዱት። እንበልና አንዳንዴ ይሄ ፎቶኮፒ ማሽን ላይ ይሄ ላይቲንጉ የሚያነበው ወደ ቀኝ ይሄዳል። ወደ ቀኝ እያነበበ ሄድና ኖቱን ሲያወጣስ በግራ በኩል እንዲህ ሊወጣ ይችላል። ኦፖዚት ዳይሬክሽን ፕሪንት ሆኖ የሚወጣው such like that the RNA polymerase is reading the RNA template towards 3' to 5' direction and undergoing synthesis in the opposite direction from 5' to 3' direction በጎን ጋር ሲንተሲስ ያካሂዳል። So unlike DNA polymerase, RNA polymerases can initiate the synthesis of new strands in the absence of a primer. ይሄን ቅድም ተነጋግረናል። RNA polymerase primary ሆነ መነሻ ሆነ RNA molecule አይፈልግም። In addition to catalyzing the polymerization of ribonucleotides, RNA polymerases must be able to recognize the sites from the parental DNA strand molecule. What are those sites? The first one, appropriate gene to be transcribed into the RNA molecules. And the appropriate strand of the double strand of DNA to copying. ምክንያቱም ሁለቱም ኮፒድ አይሆኑም RNA ሲንተሲስ ላይ RNA the single strand ሪፕሊኬሽን ላይ ችግር የለውም። ሁለቱም DNA ስትራንዶች ኮፒድ ሆነው አዲሶቹ DNA ቦዝ ስትራንድስ are synthesized. but እዚህ ላይ ግን ከሁለቱ ተመርጦ ነው appropriate የሆነው strand ተመርጦ ነው it can be copied. The selection of the appropriate strand is undergone by this RNA polymerase enzyme. And the RNA polymerase also is responsible for identifying start point of transcription. So specific sequence on DNA which can be identified by the RNA polymerase to start the transcription process is called the promoters. So it determines, the promoter region determines where the RNA polymerase binds. That is the place where it binds. And it also, the promoter region is also used for unknown the frequency of the transcription process. And it is also the site where the initiation is started for the transcription process. ሌሎች other regulatory sequences የምንላቸው አሉ እዛው parental DNA such as promoter proximal elements and enhancers የሚባሉ አሉ the gene enhancers they can be used to activate that specific process enhancing gene region የሚባሉ the promoter proximal element regions also they can act as another regulatory components for the transcription process and they also affect frequency of the transcription they may slow down or they may activate enhancers it's commonly known they activate and facilitate the transcription process and we see those regions ለምሳሌ መሃል ላይ ስታዩት እዚህ ስታርት ፖይንት አለ እንደ ዜሮ ነው የምትወሰደው toward the left side ያሉት ኒውክሊዮታይዶቹ ሲቆጠሩ ከዜሮ -1 -2 -3 እየተባለ ለምሳሌ አንዳንድ ፕሮሞተር ሪጅን ሲኩዌንስ -35 ሲኩዌንስ ሪጅን ሲባል from the starting point ወደ ኋላ 35ኛው ኒውክሊዮታይድ ሲኩዌንስ ላይ ነው ያለው ማለት ነው መጀመሪያው ያው የምንጠቅሰው ኒውክሊዮታይድ and plus 10 region ላይ ከተባለ from the start point of the transcription toward the right side it's on the 10th position ነው ማለት ነው so from starting point up to some areas you can see here that the coding region of the gene right there most of the promoters in most DNA sequence are found on the left side of the starting point of the transcription you can see here there are two boxes as acting as a promoter region and ብዙዎቹ regulatory sequences ደግሞ they are found on the 5 prime region in the region and they are found at the back of the promoter region so now a gene is a segment of DNA it contains some nucleotide sequences and that function as a unit to generate an RNA product or through transcription process RNA product forming and after that RNA product is formed it can be easily translated in order to form a polypeptide chain or protein molecule it is the last step of the central dogma of molecular biology protein synthesis the last step of the cellular response so the transcribed region of a gene contains transcribed የሚሆነው template for the synthesis of an RNA template DNA strand so which begins at the start point template has a start point the gene also includes regions of DNA that regulate production of the encoded product such as a promoter region ያቱ encoded የሚሆኑት ፕሮዳክቶች production area ያለቻቸው the parental DNA gene DNA means it contains lot of gene sequences are sequences ብዙ አሉ የተወሰኑ sequences አንድ gene ይሆናሉ so the gene also includes regions of DNA that regulate production of the encoded product so such as promoter region promoter region regulate የሚያደርግበት area አለ እኛው area as a regulatory sequence area የሚላቸው ይይዛል ማለት ነው gene and in a structural gene the transcribed region contains the first one the coding sequences that dictates the amino acid sequence of the polypeptide chain coding sequenceዎቹ ቅድም እንዳልኳችሁ ማለት promoter region ላይ ይሆናል so those sequences found in this region might dictate the amino acid sequences ማለት ነው ምክንያቱም ከነሱ የሚመጣው transcribed የሚመጣው nucleotide sequences ናቸው ኋላ ወደ amino acid የሚቀየሩት when you see in the prokaryotes in bacteria a single RNA polymerase produces the primary transcript precursors for all three major class of RNAs messenger RNA ribosomal RNA transfer RNA because they do not have specified type of subcellular organelles to undergo different process ስለዚህ primary transcript precursor-አቸው ነው እንደ ሁሉም አክት እያደረገ የሚሰራ ፓሪቶች ላይ ግን ስንመጣ specified የሆኑ separated የሆኑ RNA subunits አሉ because the bacteria do not contain the nuclei or specified የሆነ nucleus ስላልያዙ nuclear membrane ስለሌላቸው ነው the ribosomes bind to messenger RNA it is being transcribed and also in those bacteria protein synthesis occurs simultaneously with the transcription process cannot identify a specific place of subcellular organelle for prokaryotes. And eukaryotic genes are transcribed in the nucleus by 3 different RNA polymerases now, each principally responsible for one of the major classes of RNA. So these are the primary transcripts. They are modified and trimmed to produce the mature RNAs. ይሄ ቅድም የነገርኳችሁ ነው። premature RNA የሚባሉት በትራንስክሪፕሽን ፕሮሰስ ይሰራል። then after it undergoes post transcriptional modifications በዋናነት ቅድም ያልናቸው poly-A tailing እና 7-methyl guanosine capping 5' capping ያካሂዳል። ከዛ it becomes mature, it can have resistance for the nucleases, so that matured RNA can be transported to the cytosol in order to undergo further process of the translation. So the precursors of messenger RNA, they are called pre-messenger RNA, which is not activated. They have guanosine cap addition as a 5', what I told you, and poly-A tailing process ማካሄድ አለባቸው። ከዛ ውጪ ይሄ poly-A tailing እና 7-methyl guanosine capping ውጪ የተሰራው RNA contains exons as well as introns ይዟል። Exons ማለት contains the coding sequence for the proteins and are separated in the pre-messenger RNA by introns. And those introns are regions that have no coding function. ስለዚህ full functional የሆነ ለማድረግ ኢንተሮኖቹ እየተቆረጡ እየወጡ ኤግዞኖቹ አንድ ላይ መያያዝ አለባቸው። This process is called splicing process ይባላል. And there are enzyme complexes called spliceosomes which are undergoing this cutting of the introns from the middle of the mixed pre-messenger RNA, and they will then catalyze the joining up of the exons. ከዛ በኋላ ነው ይሄኛው ሶስተኛው post transcriptional modification ከሚባሉ ሶስት ነው splicing ይባላል። So during splicing reactions, introns are removed, what I told you, and exons connected to form the mature messenger RNA. In eukaryotes, transfer RNA and ribosomal RNA precursors are also modified and they trimmed, although not as extensively as pre-messenger RNA. ለምንድነው messenger RNA ላይ ነው ብዙ ጊዜ the direct genetic information can be transferred through that. So transfer RNA ላይ እና ribosomal RNA ላይ ብዙ ኢንተሮኖች ላይኖሩ ይችላሉ እንደ messenger RNA ሲታይ. So the process, even if transfer RNA and ribosomal RNA undergo modification, it cannot be like that messenger RNA. When you see the similarity and differences from the replication process again, similarity, both the replication and transcription process are undergoing synthesis in the 5' to 6' direction. In both of the processes, many proteins are involved. The other one, in prokaryotic transcription, replication, they have initiation, elongation, termination ይኖራቸዋል። ባለፈው replication ላይ እንዳየነው initiation የምንልበት step ምንድነው? The binding of the DNA polymerase in order to identify origin of replication. Initiation process ይባላል. Okay, and the binding of other proteins like single strand binding protein. ከዛ ውጭ እና ሄሊኬዝ ኢንዛይሞች ኮምፕሌክሶቹ ባይንድ ማድረጋቸው after getting the starting region, that is called the initiation complex ይባላል። formation of initiation complex initiation ይባላል። then synthesis ይካሄዳል, it's elongation, growing up of the new DNA strand. After that, whenever the required amount of DNA is synthesized, then the termination process will start. termination process ይጀምራል። እነዚህ ናቸው ስቴፖች። እዚህ ጋርም እንደዛው ነው ያለው። ሁለቱም ሶስቱም ስቴፖች አሏቸው። and the transcription bubble formation is there, replication bubble formation is complex ስላላቸው similarity ነው. And they start as the center of replication or as a center of transcription or some region, promoter region, and they stop at specific places in the required amount of the DNA or RNA synthesized. So those are similarities. When we see the differences, RNA polymerase is there instead of the DNA polymerase. RNA polymerase does not have proofreading activity for the exonuclease, 3' to 5' exonuclease. Copying activity allow 3' to 5' copying activity allow RNA polymerase. But proofreading that includes exonuclease activity of 3' to 5' doesn't have by this RNA polymerase. Then post-transcriptional modification process is taking place in here, the transcription process. However, post-replicational modification yield. And one strand copied from the parental DNA molecule in transcription process. However, replication involves the copying of both of the strands. So not all strands are copied. This is also a difference. The other difference, nucleotide triphosphates are used in the replication. However, deoxyribonucleotide triphosphates are used in the replication process. So let's transcription like the conversion of DNA space to the initiation, elongation, termination, and now. Those are three major distinct phases in most of the molecular biology parts. The first, initiation, RNA polymerase recognizes a promoter site. Promoter site means specific sequence site from the parental strand of the DNA, end of the DNA upstream to the gene, and locally unwinds the DNA to create a template. No DNA double-stranded, no RNA polymerase has unwinding activity. Promoter region ahead na DNA ይከፍተዋል እዛ ጋር ከከፈተው በኋላ ኮድ ከሚደረገው strand ላይ promoter region ላይ ሄዶ ባይንድ ይሆናል. that is the initiation process. Other proteins and enzymes are also binding that region. The elongation process involves the polymer, after once the initiation complex is formed, the polymerase starts to move along the parental gene sequences and synthesizing a complementary copy of the template DNA molecule. It synthesizes a single-stranded RNA from the template DNA, but using ribonucleotide triphosphates as a precursor. This is the total initiation, the elongation process. The termination involves when the polymerase encounters a termination sequence, it releases the transfer the RNA and dissociate from the DNA to in the transcription, and the DNA becomes rewinded. So there are different termination processes that we will see. Antiparallel complementary base pairs between DNA and RNA is formed. For instance, you can see here on the diagram, deoxyribonucleotide phosphate backbone is there, and ribose phosphate backbones for the newly synthesized RNA cells. Proofreading copying below 3' to 5' ነው የሚያደርገው RNA polymerase, because at the middle there are hydrogen bonds coming from it. Then from 5' to 3' ደግሞ synthesis እየተካሄደ ይመጣል ማለት ነው። ለምሳሌ ከዚህ እንደምታዩት DNA ነን ቴምፕሌት ወይም ኮዲንግ ስትራንድ አለ 5' to 3' ነው። ሲኩዌንሱን ተመልከቱት CG CT AT AG CG TT T ነው። ይሄ እንፈልገው ቴምፕሌት ስትራንድ ስላልሆነ RNA polymerase አይጠቀመው። but from 3' to 5' ምክንያቱም ኮፒንግ or proofreading በዚህኛው ስትራንድ ላይ ስለሚካሄድ ነው። opposite synthesis ደግሞ from 5' to 3' ስለሚሰራ ከ3' ኢንዱ እስከ 5' ያለው GCGAT ATCGCAA ነው። RNA transcription ብታዩት it is complementary with this DNA template strand. ተመልከቱ guanine is paired with what? cytosine አይደል? ከዛ cytosine with guanine እዚህ ያያችሁ ስክሪኑ ላይ guanine with cytosine adenine with uracil አይደል ምክንያቱም thiamine ስለሌለ ነው እዚህ RNA thiamine with adenine እንዲህ complementary ያደረገ the RNA transcript is complementarily synthesized from the template strand of the DNA. So DNA has template and non-template strand. Non-template strand by the way coding የሚባለው እሱ ነው. When we see RNA polymerase, usually it has 4 core subunits plus the fifth sigma factor. Because the sigma factor is responsible for determining the promoter region of from the DNA. Promoter region identify ከተደረገ በኋላ ነው ሌሎችም RNA polymerase subunits ሄደው ባይንድ ማድረግ የሚችሉት. So when we discuss about the RNA polymerase enzyme, we can call it that it has a core unit of enzyme plus sigma subunit, and both are called the holoenzyme complexes. The holoenzyme complex binds the promoter sequence region. Once the promoter sequence region is identified, it can catalyze the open complex and transcription of DNA to RNA. And this promoter region identification is performed by sigma subunit of the RNA polymerase. ለዛ ነው የሚያስፈልገው ለምሳሌ RNA bind specific promoter sequence ብለናል sigma factor recognizes consensus የምንላቸው sequences አሉ the appropriate promoter regions ለምሳሌ -10 and -35 sequences are might be acting as appropriate promoter subunits እንደምታዩት እዚህ sigma subunit አለ the remaining subunits of the RNA polymerase ቅድም ምክንያቱም holoenzyme complex ያለው the core plus sigma core subunit የምንለው it has 2 alpha subunits 2 beta subunits and beta prime subunits አለው so alpha እና alpha prime subunit beta እንዲሁም beta prime subunit አለ sigma subunit አለ እዚህ ጋር እንደምታዩት what is the main function of the sigma subunit to identify the promoter region and to lead other subunit of RNA polymerase to bind with that promoter region. The most common promoter regions እንዳልኳችሁ -10 እና -35 regions ናቸው. As I told you before, starting from the center of transcription towards the left direction, 10ኛው ኑክሊዮታይድ ያለበት እና towards the left direction -35 35ኛው ኑክሊዮታይድ ያለባቸው ሪጅኖች most promoter sequences የያዙ ናቸው. So as you can see, the consensus sequence for many prokaryotes is indicated by the E. coli promoter region. ምሳሌ messenger RNA ያለ the start site ላይ ሴንተሩ ላይ -10 region የሚባለው ብዙ ጊዜ ምን ቦክስ ይባላል መስላችሁ ታታ ቦክስ ይባላል since it contains TA ከዛ 80 ነው የሚጨምረው -10 promoter region ታታ ቦክስ ነው የሚለው -35 sequence region ያው TTGS CA ያለው ኑክሊዮታይድ ግን -35 region ይባላል after the -35 region there is an upstream element regulatory region that can be used to regulate the sequence ማለት ነው so most base substitutions in the -10 region and -35 region have a negative effect on promoter function. Some promoters also include the upstream promoter, the upstream element, promoter element. No, it can be used to regulate the sequences or the processing. And by convention, the DNA sequences are shown as they exist in the non-template strand, you know, with the 5' term minus on the left side. ምንድነው ብዙ ጊዜ የሚለየው ይሄ ቴምፕሌት እና ነን ቴምፕሌት ስትራንድ ከዲኤንኤ ሲለዩ ቅድም እንዳልኳችሁ RNA polymerase has a proofreading activity when copying activity from 3' to 5' such like that of the DNA polymerase while it undergoes synthesis from 5' to 3' direction. ስለዚህ 3' to 5' direction ያለውን ስትራንድ ነው ብዙ ጊዜ template or parental strand አድርጎ የሚወስደው. The nucleotides are numbered from the transcription start site ነው ቅድም ነግሬያቸዋለሁ ሴንተር አለች በግራ በኩል ያሉት ማይነስ እየተባሉ ነምበር ይደረጋሉ ወደ ቀኝ በኩል ያሉት ፕላስ እየተባለ ነው ነምበር ይደረጋሉ. So on this diagram, N indicates N nucleotide, the N regions. But the specific minus 35 region sequences and minus 10 region sequences are indicated. Minus 35 region sequence ከተባላችሁ TTG ACA ነው። minus 10 region sequences TAT ወይም Tata box sequence ነው። When we see the steps in RNA synthesis, the process of transcription of a typical gene, you can see in E. coli, the prokaryotes, it can be divided into 3 phases. We have seen it: initiation, elongation, and termination. And transcription unit, it extends from the promoter to termination region. The product of the process of transcription by RNA polymerase is termed the primary transcript. ምክንያቱም ፕራይመሪ ትራንስክሪፕት የምንለው ቅድም እንዳልኳችሁ ፖስት ትራንስክሪፕሽናል ሞዲፊኬሽን ስለሚያካሂድ ነው። ፕሪ ሜሴንጀር አርኤንኤ ይባላል። ፕሪ ወይም ፕሪማቹር አርኤንኤ ይባላል። in that stage. Now, when we see, as I told you before, when we see the bacterial promoters such as that of E. coli እና ዲያግራሙ ላይ በደንብ የሚመለከተው ትራንስክሪፕሽን ዩኒት አለን ቶታሊ። ሶ ስታርቲንግ ሳይት አለች +1 እያለ ፕላስ ነው የሚሄደው ራይት ሳይድ from starting site ማይነስ እያለ ሄደ። ቅድም የነገርኳችሁ -10 ሪጅን አለ ታታ ቦክስ -35 ሪጅን ዩካሪዮት ላይ TG TT GACA ሲኩዌንስ ነው ያዘ። በሌፍት ሳይድ ያለው ምንድነው? የቴምፕሌት ስትራንዱ ስታዩት የዲኑ from 3 prime to 5 prime ነው። ኮዲንግ ስትራንዱ ደግሞ የዲኒዮ ስትራንድ ነው ባይዘው እዚህ ጋር ነው። ኮዲንግ ስትራንዱ ደግሞ 5 prime to 3 prime ዳይሬክሽን ላይ ያለው ነው። ሶ ከፓረንታል ከዚህኛው ወስደን ከ3 prime to 5 prime ኮፒድ እያደረገ እነዚህ ሲኩዌንሶች ተመርቶ አዲስ ሲንተሳይዝ ይደረጋል ማለት ነው። ሶ ጂኖች ስላሉ ዲኤንኤ ላይ እዛው ጋር ነው ሬጉሌተሪ ኤሪያዎች የሚኖሩት። ሶ 5 prime to 3 prime ዳይሬክሽን RNA synthesis እየተካሄደ ይሄዳል እንደምታዩት starting from the center of the transcription from 5 prime to 3 prime አዲስ RNA so those are regions in A5 prime flanking sequence ይባላሉ። ኮዲንጉ በ5 prime region ላይ እዚህ ጋር ስላሉበት and 3 prime flanking sequences በዚህኛው ሳይድ ደግሞ ቴምፕሌት ስትራንድ ላይ እነሱ ስላሉ So initiation of the transcription process now. The process of transcription begins with the binding of RNA polymerase holoenzyme, including the sigma subunit, to a region of the DNA known as the promoter region. In the prokaryotic promoter contains characteristic consensus sequences, more probably promoter region -10 and -35 sequences. And the consensus sequences are idealized sequences in which the base shown at each position is the base most frequently. ለምሳሌ ታታ ቦክስ እና TATA ብዙ frequently በአብዛኛው ጊዜ የሚታዩ ኒውክሊዮታይድ ቤዞችን ነው እንጂ but not necessarily always. ነው especially for -35 sequence ነው። -10 ሪጅን እንኳን አብዛኛው ታታ ቦክስም የተባለው TATA ቅድሚያ የምትመጣ ነች ከዛ 8 ይጀምራል። -35 ሪጅን ግን የተለያዩ ሊኖሩት ይችላል። በአብዛኛው ግን የሚታይበት ቪታሚን ጓኒን ሪች ኤሪያ አብዛኛው ሶ ይሄ ኢኒሺዬሽን ኮምፕሌክስን ፎርም ያደርግልናል። ስቴጆቹን ብናያቸው ትራንስክሪፕሽን ኢኒሺዬሽን ፕሮሰስ ላይ ለምሳሌ RNA polymerase binds to DNA in a closed promoter complex ነው። እዚህ ጋር ፓረንታል DNA ስትራንድ አለ። ይሄ RNA polymerase including the sigma factor ነው። እቺ ሰማያዊዋ ሲግማ ፋክተር ነች። forming the ከዛ DNA ጋር ባይንድ ያደርጋል forming a closed promoter complex and this promoter complex is identified by the sigma factor. Because sigma factor allows the polymerase to convert the closed promoter complex to an open promoter complex. As you can see on this page, formation of the open promoter complex comes there. Once open promoter complex is formed, the polymerase incorporates first 9 or 10 nucleotides into the nascent RNA. RNA polymerase በራሱ እዚህ 9 or 10 nucleotide sequence they are acting as nascent ወይም የመጀማሪ RNA ሞለኪውሉን ያገለግሉናል እነሱ ላይ ያደገ ይዳል ግሮዊንግ ቼን DNA polymerase ከዚህ በፊት እንዳልነው ፕራይሜዝ አማካኝነት እነዚህን የተወሰኑ RNA ስትራንዶችን አምጥቶ እንደ ፕራይመሪ ይጠቀማል RNA polymerase በራሱ ነው የሚያመርተው nascent RNA the polymerase clears the promoter region identified and after identification of the promoter region sigma subunit of the RNA polymerase will be lost from the other subunits and begins the elongation process. Losing of this the sigma subunit of RNA ያሉት አራት ስቴፖች elongation እስከሚጀምርበት ያለው step is called initiation of the transcription process ይባላል. And the sigma stimulates the initiation but not elongation. Because ቅድም እንዳልኳችሁ sigma subunit follow enzyme complex it comes the major function of that is to identify the promoter region for the remaining subunits of the enzyme. So double helical DNA ያለ unwinding ይፈጠራል. Means DNA unwinding is undergone by the RNA polymerase and RNA polymerase is undergone unwinding of around 17 base pairs of the template DNA in order to initiate and start the process. The unwinding would occur because as the copying is taking place from the parental DNA, even DNA synthesis like the unwinding is common to synthesize. And one of the basic feature of both RNA polymerase and DNA polymerases, they undergo copying from 3' to 5' direction from parental strand, and they undergo synthesis from 5' to 3' synthesis direction. This diagram ላይ እንዳያችሁት ፈዘዝ ባለ greenish color እና deep greenish color. It can be used to indicate the presence of introns and exons in the synthesized RNA molecule. So, chain ላይ ሁሉ ጊዜ unwinding ሲፈጽሙ parental DNA strand ላይ ከኋላ ጋር እዚህ ጋር unwinding አለ እንደምታዩት እንዲህ ሲመጣ this side on the right side ማለት ከናንተ በኩል unwinding አለ እየተፈታ ነው የሚመጣው ነው እዚህ ጋር ሲገባ ሙሉ ለሙሉ ይፈታ. After that, after the synthesis taking place, the rewinding is also performed by the RNA polymerase enzyme. By the way, this type of RNA polymerase enzyme is called DNA-dependent RNA polymerase, as it uses DNA as parental strand. RNA-dependent RNA polymerase whichም አሉ. እንደገና DNA-dependent DNA polymerase አለ. The replication process ላይ እንደገና RNA dependent DNA polymerase አለ reverse transcription process ላይ ቫይረሶች ነው ብዙ አክቲቪቲ ያላቸው RNA dependent DNA polymerase activity especially HIV ቫይረስ አሁን elongation ላይ ስንመጣ it is called when the transcript typically starting with purine no exceeds 10 nucleotides in length ነው ቅድም ኢኒሺዬሽኑ ላይ እንዳየነው እስከ 10ኛው ኑክሊዮታይድ ድረስ it's acting as the nascent RNA ነው it can be used as a starting for the elongation of the elongation process እስከ 10 ድረስ ከሞላ በኋላ ያለው after that the sigma subunit will be released ከዛ ቀጥሎ ያለው ስቴፕ ነው elongation የሚባለው sigma subunit of RNA polymerase should be released in order to start the elongation process. Sigma is then released, the core enzyme is able to leave the promoter and move along the template strand in a processive manner. So during transcription, a short DNA RNA hybrid helix is formed until the elongation process is terminated. So like DNA polymerase, RNA polymerase uses nucleoside triphosphates as substrate. DNA polymerase ያው deoxyribonucleotide triphosphates ነው የሚጠቀመው nucleotides ነው ቅድም ብለናል and it releases pyrophosphate ነው each time and the nucleoside monophosphate is added to the growing chain. The breakdown of pyrophosphate provides energy from that nucleotide triphosphates. When the breakdown of pyrophosphates from the nucleotide triphosphates provides energy for the formation of phosphates or phosphodiester linkage on the growing chain of synthesized RNA. So like replication, transcription is always the 5' to 3' direction. Zigam diagrammatically, the DNA RNA hybrid or helix can be formed in here. And this is a polymerase, no? RNA polymerase enzyme. After the removal of this sigma subunit, sorry, sigma subunit ለብቻው ስላለ ነው unwinding activity ያለው ብለናል and rewinding activity ያለው the coding strand ተነጠለበት means and template strand ብዙ ጊዜ template strand አልኳችሁ 3' to 5' ያለው አማራጭ የሚወሰደው based on the template strand ታያችሁት ከtemplate strand ላይ RNA synthesize ማለት ነው 5' to 3' direction transcription bubble እንለዋለን እንግዲህ ነው transcription bubble schematic representation of the transcription bubble in the elongation of an RNA transcript. Transcription bubble has a difference during the stage of initiation and during the stage of elongation. Elongation why one of the reasons is during initiation there is the presence of sigma subunit of the enzyme. During elongation already the sigma subunit of the enzyme is left. And duplex DNA is unwound at the forward end, in the left side direction, the right side direction, in each side. And it can be rewinded after the process is taking place. The RNA DNA hybrid rotates during the elongation. የሚካሄደው ፕሮሰሱን እየተሽከረከረ ኢንዛይም ባይንድ ሆኖ ፕሮሰሱ ይካሄዳል። So unlike DNA polymerase, RNA polymerase does not require a primer ብለናል ብዙ ጊዜ ነው ያለው and has no non-proofreading activity of with regards to exonuclease. Copying ከሆነ አለው activity. It's not worthy that RNA polymerase lacks nuclease activity. It doesn't have. Thus, in contrast with DNA polymerase, RNA polymerase does not correct the nascent polynucleotide state. ከዚህ በፊት DNA polymerase ምን አይነት አክቲቪቲዎች አሉት ብለናል? exonuclease activity, endonuclease activity, special DNA polymerase 3, DNA polymerase 1 አሉ ብለናል። Even if DNA polymerase 1 is a major polymerizing enzyme as it has highest speed of polymerization in comparison with DNA polymerase 1 and DNA polymerase 2. Higher polymerizing ስላለው major polymerizing enzyme ነው ብለናል ሬቱ ከፍተኛ ስለሆነ and DNA polymerase 1 ደግሞ exonuclease activity ያለው nascent synthesized DNA ስትራንድ ላይ ያሉ ስህተቶችን ሁሉ ማስተካከል ይችላል። nuclease activity ስለሌለው ነው RNA polymerase እነዚህ ነገሮች ማድረግ የማይችለው። በዚህ DNA polymerase have that nuclease activity ስለዚህ ያስተካክላል። Consequently, fidelity of transcription is much lower than that of the replication as I told you. ቅድም ያልኳችሁ replication በጣም fidelity ያለው ለዛው ነው ዓለም ላይ ያለ ነው ብዙዎች ሰዎች ኖርማል የሆነ ፊዚካል ስታተስ እና ጄኔቲካል ስታተስ ያለው ተወሰነ ነው ኢራር የሚፈጠረው fidelity ለትራንስክሪፕሽን ላይ ግን ስንመጣ እንደ DNA polymerase ያህል perfection አይኖርም. And the error rate of RNA synthesis is of the order of one mistake per 10⁻⁴ or 10⁵ nucleotides. ባለፈው DNA replication ላይ ያየነው per 10⁻17 ነው ያለው 1 ሊከሰት ይችላል. About 10⁵ times as high as that of DNA synthesis. Error rate formation now እዚህ ላይ ከፍ ይላል ማለት ነው. the much lower fidelity of RNA synthesis can be tolerated because the mistakes are not transmitted to progeny. ወደዛ ሊተላለፉ ስለማይችሉ ዲኤንኤው ላይ ግን ቢሆን ኖሮ እንደ ፓረንታል ስትራንድ የሚያገለግለው ቴምፕሌት ስትራንድ የሚያገለግለው ስለሆነ እየተላለፈ ነበር የሚሄደው አማራጭ አይኖርም ነበር. So for most genes, many RNA transcripts are synthesized. A few defective transcripts are unlikely to be harmful. Out of that, the remaining cannot be active as harmful because since they all not may be undergone translation process or synthesis of protein. Termination ላይ ስንመጣ the termination of transcription is as precisely controlled as its initiation. And the elongation of single-stranded RNA chain continues until the termination signal is reached to that point. In the termination phase of transcription, the formation of phosphodiester bonds ceases. RNA DNA hybrid dissociate ያደርጋል termination process ላይ the melted region of DNA ወይም unwinded region ነው የምንለው ድጋሚ rewinded ይሆናል ከዛ ውጭ RNA polymerase released from the DNA ነው DNA strand ላይ released ያደርጋል እነዚህን አንድ ላይ ያቀፈው process is called the termination እንለዋለን in termination there are different types of termination majorly ሁለት ነው እንግዲህ row dependent እና row independent የሚባሉት row independent የሚባለው it's an intrinsic spontaneous termination process no without participation of external things. But row dependent row is a protein. It's an external protein. It comes to the during transcription process. It comes to the region in order to initiate the termination of the synthesis of the transcript. So when we see that an inverted repeat base sequence containing a central non-repeating segment አለ እዚህ ጋር ለምሳሌ inverted repetition አለ ሌላኛው a sequence having a high G+ C content አለ G+ C ኮንተንቶችን ይይዛል so sequence adenine thymine pairs in DNA may begin in the stem and that results in a sequence of 6 up to 8 uridine in the RNA ነው 6 up to 8 uridine እቺ መጨረሻ 3 prime end ላይ ዲያግራሙ ላይ የምታዩት በሚመጣበት ጊዜ we can call it that it is an intrinsic termination process signaling እንለዋለን polyuninucleotide sequences when they come to the 3 prime end እነዚህ መጡ ማለት ምን ይመሰረታል ይሄ የምታዩት hairpin structure እየተጓዘ ከነበር ቀጥ ብሎ የሚያልፍ ከነበር ለምሳሌ ወደ ላይ ለሁለት ታጥፎ ኒውክሊዮታይድ ቼኖች በሚመሰርቱበት ጊዜ እየተንሸራተተ መሄድ አይችልም ከዛ በኋላ hairpin form አድርጎ ስለዚህ the synthesized RNA becomes separated from the parental DNA as it cannot be undergo movement in such a condition so ይሄ hairpin formation ለማቆም ወይም እንበላችሁና የሆነ ነገር ያሳለፋችሁ ሽቦ ለምሳሌ ቀጥ ብሎ የሚያልፍበት ማሽን እንደዚህ ካለ በማሽን ላይ ያለፈ ከሆነ ታጥፎ ለሁለት እንደዚህ hairpin structure ቢሰራ ምንድነው የሚሆነው ይቆማል ከዛ በኋላ ሊያልፍ አይችልም ይሄኛው ወደ ኋላ መልሶት ሊወጣ ይችላል so the base sequence of the DNA of E. coli there are operons የምንላቸው አሉ ባይዘው one of the operon is tryptophan operon it can be used to regulate termination ላይ ነው E. coli prokaryotes ላይ ሲሆን at which transcription termination occurs end of ነው ነገር ግን 3 terminus end of the messenger RNA molecule of the eukaryotes ላይ ግን polyutailing can act as termination signal ሆኖ ያገለግለናል hairpin formation ላይ termination of transcription includes the following steps ነው that means intrinsic ያለው it's not row dependent row dependent ሲሆን ሌላ ተጨማሪ ፕሮቲን ሮ የሚባል መጥቶ ነው termination የሚፈጠር የሚያደርገው the intrinsic ላይ ቅድም እንዳልኳችሁ cessation of RNA elongation ይኖራል ይቆማል in the polyutailing can act as ምክንያቱም polyutailing ከመጣበት ጎን ጋር hairpin structure that is responsible for the termination is formed. A release of newly formed RNA ይኖራል። Release of RNA polymerase from the DNA parental strand ከዚያ መጨረሻ ላይ dissociation of the transcription complex ይመጣል ማለት ነው። And termination signal is found at, as I told you, the 3' end of the messenger RNA transcript, an intrinsic termination signal. Okay, it consists of series of bases that form a stable stem loop structure and series of uridine residues. Loop structure ምንላት ይችላል? loop አይነት structure, hairpin structure ምንላታለን። This structure is formed with a pairing of nucleotides on the same strand of this RNA. So responsible ይሆናል። intrinsic ላይ ሲሆን extrinsic is row dependent ይባላል። row dependent ነው የሚባለው extrinsic. So still intrinsic ላይ ስለሆነ model for intrinsic termination ነው የምናየው የተካሄደ ነበር። The polymerase has posed at a steering of weak uridine ribo-uridine sequences ላይ እና ዲኦክሲአዲኔዚን ብዙ ጊዜ ከሜሴንጀር አርኤንኤ ነው ከሲንተሳይዙ ብዙ ዩሪዲኖች ሲመጡ ከፓረንታል ዲኤንኤው ደግሞ አዲኖዚን ያቱም ዘ ፎርምስ ፔር ነው የሚያሳዩት ደብል ቦንድ ስለሆነ በሁለቱ መካከል የሚፈጠረው ፔር ዊክ ነው ሊንኬጁ እዚህ ቦታ ላይ ያቱም ቤንዲንግ ለመፈጠር ያገለግላል ኢንትሪንሲክ ቤንዲንግ ወይ ሄልፒንግ ፎርም in the hairpin form has started to form just downstream of the person. የዚህ hairpin structure እየተፈጠረ መሄድ it cannot allow so once the hairpin is formed it destabilizes the hybridization of RNA and DNA. After the destabilization process, as you can see on the screen, the formed synthesized RNA will be separated from parental DNA. After that, the enzyme will also leave that complex, the termination will take place in DNA. ተመልሶ circular structure. So as the hairpin forms, it further destabilizes RNA DNA hybrid, and this destabilization could take several forms. Now, the formation of the hairpin could physically pull the RNA out of the polymerase. በራሱ እዚህ ጋር ጫና ስለይበዛበት ክብደት ስቡ RNA ሊያወጣው ይችላል። ሁለተኛው it could simply cause the transcription bubble to collapse ነው። ይሄ መፈጠሩ hairpin structure መፈጠሩ በራሱ ትራንስክሪፕሽን በብሉን ኮላፕስ እንዲያደርግ ያደርገዋል በዚህ ጊዜ RNA ይወጣል ከዛ RNA polymerase separated ይሆናል ማለት ነው so the RNA product in polymerase dissociates completely from the DNA template and that means what you call it the termination of the transcription can be taking place this is an intrinsic termination extrinsic ሲሆን ግን bending or hairpin structure formation on ማምጣት የሚችለው ማን ነው external factor that means a protein what you call it that zero protein raw affects chain elongation but not initiation raw protein initiation ቅድም ያየነው ስለሆነ the raw causes production of shorter transcripts and raw seems not to have affinity for RNA polymerase and whereas the sigma is critical part of the polymerase enzyme complex raw is not critical part of the enzyme complex. That's why we call it rho is a protein and it's an extrinsic factor to create this termination of transcription. So many are binding of the rho to the RNA activates the ATPasm. So rho movement አለ በአርኤንኤ ላይ በሚሄድበት ጊዜ rho ፕሮቲኑ እንቅስቃሴ አለው ያ እንቅስቃሴው ማጣፍን ነው የሚያመጣው hairpin structure formation ሊመጣ RNA ማጣፍ ነው የሚያመጣው ያ እንቅስቃሴ ደግሞ ስላለ ሮ ፕሮቲን has ATPase activity ይኖራል breakdown of ATP ያካሂዳል in order to undergo binding which supplies the breakdown of ATP supplies energy to propel the raw hexamer along the RNA to move the raw hexamer along the RNA from 5' to 3' direction following the RNA polymerase activity direction in order to form that hairpin structure by following that RNA polymerase. And this chase continues until the polymerase stalls in the terminator region, just after making the RNA hairpin. Then the row can catch up and release the transcript. The row protein by itself can catch up. As you can see here, the model. So this is a row protein. Row protein should undergo a movement from 5' to 3' direction, because as you have, it has an ATPase activity. It gets energy from the ATP. So row binds to transcript at row leading site and pursues polymerase. Polymerase በሚጓዝ የፖሊመሬዙን አክቲቪቲ እየተከተለ ነው የሚሄደው በሱ ዳይሬክሽን from 5' to 3' direction። ከዛ እየተከተለ ወደ ላይ እየመጣ ሄርፒን ስትራክቸሯ መጥፋቷ እንድትፈጠር ነው የሚያደርገው። ሄርፒን forms and polymerase pauses due to the hairpin formation structure, and row catches up that hairpin structure. Hairpin structure ጋር catch up አድርጎ ይቆይና እንዲሳብ ነው የሚያደርገው። ያ ተስቦ ወጣ ማለት ከወጣ በኋላ the transcription complex becomes dissociated ነው። ሮም ለብቻ ይወጣል። synthesized RNA will be released ማለት ነው። So the polymerase makes RNA, row binds to transcript at a row leading site and pursues the polymerase, and the hairpin forms in the transcript. Then the polymerase pauses due to the formation of hairpin, and giving row a chance to catch up it and to release it from the transcription complex ማለት ነው። So ኋላ ላይ ምንድነው መጨረሻ ላይ row helicase አለ። It unwinds the RNA hybrid and release the transcript ማለት ነው። helicase ባለፈው እንዳየነው undergoes unwinding process during the DNA replication ብለናል። እዚህ ጋር helicase activity ያለው ፕሮቲን ስለሆነ row helicase, all enzymes are proteins except RNA. Row protein has an helicase activity. በዛ ምክንያት unwind ለማድረግ ያገለግላል፣ ለማላቀቅ ማናማንን RNA synthesized RNA from the parental DNA strand ማለት ነው። So the row dependent terminators consists of ያልኳችሁ an inverted repeat is there, and which can cause a hairpin to form in the transcript. As I told you, that the hairpin structure is formed mostly in the area of there is a linkage between DNA and RNA that is rich in uracil and adenine. Adenine from the RNA strand and uracil from the RNA synthesized template. So this shows mechanism for the termination of transcription by the row protein, and binding of the row protein, as you can see here, will undergo a movement on the direction of the RNA polymerase. And for a movement, it requires energy, and that energy is obtained from the breakdown of ATP. The ATP is broken down into ADP plus PI ማለት ነው. So row is an external protein factor that undergoes the termination of transcription. ሁለቱንም አየን ማለት ነው። row independent as well as row dependent termination of protein synthesis. When we see action of some antibiotic drugs, they prevent bacterial cell growth by inhibiting RNA synthesis of the bacteria. For example, we can see that rifampin, it is an inhibitor. It inhibits the initiation of transcription by binding to the beta subunit of prokaryotic RNA polymerase. Even if sigma is acting as a detector for the promoter region, if the beta subunit is blocked, it cannot be, the sigma subunit cannot be combined with the whole enzyme complex and cannot undergo the further process. So rifampin is acting as an inhibitor. It's interfering with the formation of the first phosphodiester bond formation. No? Then rifampin is useful in the treatment of tuberculosis. ብዙውን ጊዜ በዚህ መልኩ tuberculosis bacterial growth ለመግታት ነው የሚያገለግለን። Another one, dactinomycin. It's known to biochemists ላይ ብዙ ጊዜ actinomycin D ይባላል። which was the first antibiotic to find therapeutic application in the tumor chemotherapy. The tumor cells line. Now, binds to the DNA template structure and interferes with the movement of RNA polymerase along the DNA. That means it blocks the RNA polymerase from undergoing movement on the DNA strand. If it cannot undergo the movement, means the copying cannot be taking place. If the copying is not been taking place, means the new synthesis of RNA cannot be taking place ማለት ነው በሌላ መንገድ። ስለዚህ በዚህ መልኩ ብሎክ ያደርጋል። ለምሳሌ እዚህ ጋር እንደምታዩት inactivation of prokaryotic RNA polymerase by rifampin ላይ ስታዩት አሁን ኖርማል ድረግ በሌለበት ሁኔታ ቀደም እንዳየነው RNA polymerase RNA DNA hybrid formation አለ። polymerase unit bind ያደርጋል. The elongation process will take place. However, when the rifampin is binded with the RNA polymerase, the RNA polymerase will not undergo further movement. So we can call it that rifampin binds to RNA polymerase and changes its conformation so that it cannot initiate RNA synthesis, cannot undergo movement to undergo the further process of synthesis, and the RNA polymerase from eukaryotic cells does not bind rifampin. And RNA synthesis is unaffected in the eukaryotic cells because there is a difference between the prokaryotic and eukaryotic cells. እኛ ሰውነት ውስጥ አሁን ለምሳሌ መድኃኒቱን ስንወስደው ታርጌትፉሊ ሄዶ RNA polymerase ኢንሂቢት ሊያደርግ የሚችለው የፕሮካሪዮቶች ወይ ቱበርኩሎሲስ ላይ ያሉት ባክቴሪያስ እና በዚህ መልኩ እነሱን ብቻ አታክ በማድረግ ባክቴሪያዎችን ያዳክማል። The transcription of eukaryotic genes, when we can see that the transcription of eukaryotic genes is a far more complicated process than its transcription of eukaryotes. It's common, and one of the reasons include eukaryotic transcription involves separate polymerases, and also those different types of polymerases are involving in the synthesis of ribosomal RNA, transfer RNA, and messenger RNA. We say that in the eukaryotic synthesis of RNA, the process is somehow different from the eukaryotes because the prokaryotes doesn't have specific subcellular organelle membranes, and they have one general synthesized RNA molecule in order to indicate the ribosomal RNA, transfer RNA, and messenger RNA. But for the eukaryotes, each of those types of RNA should be synthesized as they do have their own function in various subcellular organelles in nucleus and in the cytoplasm. So they do have various functions. So in addition, number of proteins called transcription factors are included or involved in the transcription process of eukaryotes. So the transcription factors, they bind to distinct sites on the DNA, and either within the promoter region of the DNA or some distance from it, in eukaryotic cells. And they are required both for the first one, the assembly of transcription complex as a promoter region, that means transcription bubble formation ላይ ያስፈልጉናል። ሌላው the determination of which genes are to be transcribed, or what you call it, identification of the promoter regions of the genes from the DNA parental strand. So for the transcription factors to recognize and bind to the specific DNA sequences, those additional protein factors are required. ከዛ ውጪ the chromatin structure in the region must altered to allow access to the DNA. Prokaryotic ላይ ሲሆን they do not have histone proteinoች now with chromatin regions. But in the prokaryotes there are histone proteins which can be used to complex with the DNA in order to keep it in the original form, in the processive form ነው። So each eukaryotic RNA polymerase has its own promoters and transcription factor regions in opposite to the prokaryotic RNA polymerase. As you can see here on the structure, the naked RNA strand is there, may not be complexed with the histones. However, in order to keep the naked DNA structure is a double strand. In order to keep this DNA structure in a chromatin, okay, our body needs to have their inner complex form with the histone proteins. So it undergoes the histone binding, as you can see here. Okay, histone proteins they form a link ማለት ነው። እዚህ ዲያግራሙ ላይ እንደምታዩት then chromatin ላይ ለማስቀመጥ ይጠቅማል። In the second process is nucleosome and the beads histone plus DNA can be formed. Then nucleofilaments can be formed. They have a fiber structure. Finally, nucleofilaments they are coiled up together and anchored to scaffold the protein in order to form the chromosomes. Chromatin structure and the gene expression. They are most actively transcribed genes are found in this chromatin structure. And also relatively relaxed form of chromatin called euchromatin is there. And the euchromatin, most inactive segments of the DNA are found in the highly condensed heterochromatin form. ከዛ በኋላ ነው እንግዲህ the association of DNA with histones can be formed in order to form nucleosomes. And it affects the ability of transcription machinery to access the DNA to be transcribed. ስለዚህ በዚህ ፕሮሰስ ላይ ምን መሆን አለበት ማለት ነው? There should be different types of RNA polymerases in the eukaryotes to undergo the different synthesis of ribosomal RNA, messenger RNA, then transfer RNA. And the interconversion of active and inactive forms of the chromatin is called chromatin remodeling ይባላል። እሱን እናየዋለን። Chromatin remodeling is mostly formed through acetylation of lysine residues in the amino terminus of histone proteins. ለምሳሌ what you call is that histone acetylation and histone deacetylation process ነው የምንለው activation and deactivation. As you know that the proteins have amino end and carboxyl እንዳላቸው. The histones are proteins. If the histones are undergone acetylation process, the free amino end of them can participate in the bond formation with the acetyl group, and it cannot be combined with the DNA in order to form DNA histone complex formation. So, the chromatin remodeling ጊዜ if the histones are acetylated means they are not ready to bind with the DNA ማለት ነው። DNA free ይሆናል ማለት ነው። If the histones are undergoing deacetylation by the histone deacetylase enzyme activity, they will create the free amino end of the DNA to form a bond with the negatively charged end of the free DNA strand. ከዛ DNA is complexed with the histone እንላለን። በዛ ጊዜ we cannot get free DNA for undergoing the replication process or for undergoing the transcription process because the DNA is a parental strand. So acetylation or deacetylation of a lysine residue in a histone protein can be taken because as you can see on the diagram, the lysine provides free amino end for the proteins that we have for the histone proteins. The activation and inactivation is taking place through acetylation and deacetylation of the residues. When you see the acetylation, as I told you before, the acetylation eliminates the positive charge on the lysine residue of the histone proteins. That means it decreases the interaction of the histone with the negatively charged DNA strand in this process of opening up the chromatin and making DNA more accessible for the transcription process as well as for the replication process also. And this reaction is mediated by the catalyzed activity of histone acetyl transferases, and the deactivation process of the DNA strand for not undergoing this transcription process is catalyzed by the histone deacetylases, and those histone deacetylases removes the acetyl group from the histone, the lysine residue of the histone protein, and they restore the positively charged of the histone lysine end, and it can have a conjugation with the DNA in order to form a complex DNA, and it will be kept in the chromosome form, in the chromosomes. So it fosters a stronger interaction between histones and the DNA. When we see the nuclear RNA polymerase of eukaryotic cells, there are three distinct classes of RNA polymerases in the nucleus of eukaryotic cell. And they all are large enzymes with multiple subunits. RNA polymerase 1 is there, which is found in the nucleolus now. Nucleolus ላይ ብዙ ጊዜ ባለፈው the region where the DNA replication and RNA transcription is taking place. And the other two polymerases are located in the nucleoplasm. And the three nuclear RNA polymerases have different role in the transcription in eukaryotic cells. Each class of RNA polymerase recognizes particular type of the genes which are present in the parental DNA strand. When we see the RNA polymerase 1, it is found in the nucleolus, I told you. And the polymerase 1 makes a precursor of 5.8S, 18S, and 28S ribosomal RNAs. As we discussed in the ribosomes part, eukaryotic ribosomal RNA has the subunits, the larger subunit and smaller subunit. Those subunits have the sedimentation coefficient which አሏቸው ለራይቦዞም አርኤንኤውን በዛ መልኩ ሜጀር ስለሚደረግ so 5.8S 18.8S 18S sorry and 28S ribosomal RNAs they can be synthesized by this polymerase 1 activity factors ማለት polymerase 1 እነዚህን ribosomal RNAዎችን synthesize ለማድረግ ያገለግላል ማለት ነው። So for RNA polymerase 1 there are promoter regions, you can call them class 1 promoters ነው የምንላቸው. Those class 1 promoters, they are not well conserved in sequence from one species to another species, but the general architecture of the promoter is well conserved. That when you see that architecture, it consists of two elements. The first one is a core element. This core element is surrounding the transcription start site, the center site, and also, as we have seen previously, there are upstream control element regions in some of those genes. Those upstream control elements are about 100 base pair farther from the upstream region itself. And the spacing between the two elements is important. As you can see here on the diagram, the two ribosomal RNA promoter elements of human ribosomal RNA gene are indicated. The starting site, what you call it, it's the zero site, center ስለሆነ toward the right side ኒውክሊዮታይዶችን ስንቆጥር +1 +2 +3 +4 እያለ ነው የምንሄደው. So core regional on eukaryotic ribosomal RNA synthesis ላይ ጂኑ ኮር ሪጅኑ ከ-45 እስከ +20 ማለት ያሉት ኒውክሊዮታይዶች ይዛል ኮር ሪጅን and upstream control element region እንደምታዩት from -107 to -156 region area ላይ ነው ያለው. So the promoter has two important regions as you can see on the diagram, the upstream control element region and the core region አለ. The upstream control element is necessary for optimal transcription process. It can be used to control, but not for basal transcription. It cannot control the basal transcription process. ያ ምንድነው? The basal transcription is possible in the absence of upstream control element ማለት ነው. The optimal transcription cannot be possible without it. And the core element is absolutely required for any transcription to occur now. ምክንያቱም the core element region there are genes that are specific for any kind of absolute the transcription process. When you see RNA polymerase 2, the RNA polymerase 2 synthesizes heteronuclear RNAs, heteronuclear RNAs, and which are precursors to the messenger RNA, the synthesis process. So because, as we said before, each of RNA polymerases in the eukaryotic genes have their own function to synthesize a specific type of RNA. And this the RNA polymerase 2 also makes most smaller nuclear RNAs. Small nuclear RNAs, heteronuclear RNAs are there. But one of the advantages is that in the eukaryotic cells, those RNAs might be premature RNAs and they might undergo post-transcriptional modifications. This enzyme can recognize thousands of promoters that vary greatly in sequence. RNA polymerase 2 enzyme gene regionዎችን ብናያቸው parental DNA strand ላይ from the center +1 region ላይ በቀኝ በኩል downstream element ይኖራል internal region ይኖራል or -25 sequence ቅድም ያለው tata box might be there. Then ከታታ ቦክስ በግራ በኩል ወይም ቀድሞ የሚመጣው upper stream element region ነው towards the left side of the tata box -25 region. So generic polymerase 2 promoter regions seems like this one. They contain upstream element region, tata box, internal region ይኖራል to start around the starting point of transcription and downstream elements አሉት ማለት ነው. Downstream element after the starting point of the transcription ነው የሚኖረው. So the class 2 promoter regions because RNA polymerase 2 ካለ የclass 2 promoter regions የምንላቸው ነው ዲስከስ ማድረግ አለብን. Those regions may contain any of the following elements. Tata box, an upstream element. An upstream element might contain GC box ነው guanosine, cytosine, nucleotide rich box ሊይዝ ይችላል or it might be also CCAAT box region ሊሆንም ይችላል. class 2 promoter region initiator ይኖረዋል ቅድም ዲያግራሙ ላይ እንዳየነው internal starting point region ላይ ያለው initiator region አለው and the downstream element region as you can see it is found on the the top right side of the parental strand of such a model. So at least one of those elements is missing in many promoters. And the promoters for highly expressed specialized genes, they tend to have tata boxes. RNA polymerase 2 promoter region line. RNA polymerase 1 region ተብላችሁ ብትጠየቁ on which one of the following regions are absent in the RNA polymerase 1 gene ብትባሉ ለምሳሌ ታታ ቦክስ እዛ ጋር ኢንዲኬትድ አልሆነ ኮር እና አፕስትሪም ኮንትሮል ኤሌመንት ነው ከዛ ውጪ ኢኒሺየተር region is not indicated of the gene region of RNA polymerase 1. So rather than that, the promoters for housekeeping genes tend to lack them. And some promoters have conserved initiators and possibly with the downstream element in the class 2 promoter regions. And most genes have some kind of upstream element and many have more than one upstream element. In the model diagram that I indicated here, it has one upstream element in yellow. ነገር ግን ሌሎቹ RNA polymerase 2 gene sites might have more than one upstream element regions to ስሪም ሊኖራቸው ይችላል ማለት ነው። when you see the RNA polymerase 3 makes the precursors to 5S ribosomal RNA in the transfer RNA. Several other small cellular and viral RNAs are there. እነዚህን ይሰራልናል። እዚህ ጋር ስፔሲፊካሊ ማወቅ አለባችሁ RNA polymerase 3 ካሪየስ ላይ የትኛዎቹን ታይፕስ ኦፍ ራይቦሶማል አርኤንኤ ይሰራል በል 5S rather than the transfer RNAs ቅድም እንዳየነው RNA polymerase 3 የትኛዎቹን ታይፕ ኦፍ RNAዎችን ለማምረት ነው የሚያገለግለው ስንል ሄትሮኒኩላር RNAs እና ሜሴንጀር RNA ከዛ በፊት ደግሞ ወደ ላይ መተን እዚህ ራይቦሶማል RNA የትኛዎቹን ታይፕስ ኦፍ RNAዎችን ለመስራት ያገለግለናል ስንል 5.8S 18S and 28S ribosomal RNAዎችን ለመስራት ነው የሚያገለግለን. That's why we are saying each of the eukaryotic RNA polymerases have their own specific function to synthesize specific types of RNAs. ለዛም የራሳቸው የሆነ ፕሮሞተር ሪጅን ያላቸው የራሳቸው የሆነ ሪጅኖች ባይንዲንግ ሪጅኖቻቸውን the parental DNA strand ላይ ስፔሲፊክ የሆኑት RNA polymerase 3 the 5S ribosomal RNA እና transfer RNA synthesize ለማድረግ ያገለግለናል. And as I told you before, there are several small cellular and viral RNAs can be synthesized by the activity of this enzyme. RNA polymerase 3 transcribes a variety of genes that encode small RNAs. And those smaller RNAs include the first one, what you call it, the classical class 3 genes የምንላቸው classical class 3 genes ነው ኤሪያዎች ለRNA polymerase 3 ስለሆነ class 3 genes ናቸው. They including the 5S ribosomal RNA and transfer RNA genes ናቸው በፈርስት ክላስ ላይ and the adenovirus ወይም viral RNA genes የምንላቸው በዚህ classical class 3 genes ውስጥ ይካተታሉ. Those have promoters located entirely within the genes themselves ነው. ከጂን አንዳንዶቹ ላይ ምክንያቱም ከጂኑ ውጭ ያሉ ፕሮሞተሮች ያሏቸው አሉ። አሁን ለምሳሌ ነን ክላሲካል ክላስ 3 ጂንስ የምንላቸው ሁለተኛው by the way all those genes are working for the RNA polymerase 3 በዛ ነው እነዚህ ላይ ነው እንትን ያለው RNA polymerase 3 transcribes a variety of genes ስላሉ that encodes small RNA እዛ ነው እነዚህ ጂኖች ነው ያየነው ክላስ 3 classical class 3 genes አየናቸው. They include the genes include the site for synthesis of 5S ribosomal RNA and transfer RNA genes ብለናል. እዛዎች አዲኖ viral RNA genes ይካተታሉ ብለናል. They are promoters located entirely within the genes themselves ብለናል. ሁለተኛዎቹ non classical class 3 genes ስናይ they are relatively recently discovered class 3 genes ናቸው. ከሌሎች ከነዛ ከክላሲካል class 3 genes ጋር ስናያቸው they include the U6 small nuclear RNA የምንላቸው genes አሉ. እነዛን ጂኖች ይዘዋል. the U6 small nuclear RNA ሲንተሲስ የሚያገለግሉ ጂኖች the 7SL the 7SL RNA gene ይዘዋል. እነዚህ classical class 3 classical non-classical gene regions. They also contain the 7SK RNA gene and the Epstein-Barr virus ለዛም በቅርብ ጊዜ የተገኘ Epstein-Barr virus ጂን አለ. ያኛው ጂን ማናቸው EBER2 ጂን ይባላል. እነዚህን ክላስ 3 ጂንስ ላይ የሚካተቱ ናቸው. They are promoters that resemble those found in class 2 genes ነው. ምክንያቱም ክላስ 2 ጂንስ ላይ ካየን ፕሮሞተር ሪጅኖቻቸው እዛው አካባቢ ላይ ነው የሚገኙት ብለን እዛው ዲኤንኤ ስትራንድ ላይ ለምሳሌ ያው አሁን RNA polymerase 2 ክላስ 2 ጂንስ አሉት ፕሮሞተር ሪጅኑ እሺ class regions ላይ እዚህ ጋር ነው ዲያግራሙ ላይ ያለው promoters of some polymerase regions አሉን ለምሳሌ intermediate element ይኖረናል ቦክሶች ይኖራሉ 5 prime end ነው this is a 3 prime end the box might be box A might contain one specific promoter box C ነው intermediate element መሃል ላይ ይኖራል ትራንስፈር አርኤንኤ እና ደግሞ ቫይራል አርኤንኤ ላይም አለ የነዛ ላይ ያለው ሞዴል ጂኑ ምክንያቱም እዚህ ጋር ተከስተናል ክላሲካሊ ክላስ ሪጅንስ ክላሲካሎቹ ማለት ነው ይሄ ሁለተኛው እሱ ነው የሚያሳየው ክላሲካል ሪጅንስ አንደኛው ደግሞ ክላስ 3 ሆኖ ነን ክላሲካል ሪጅንስ ኢንዲኬተር ማለት ነው so the promoters of the 5s transfer RNA and viral RNA genes are depicted as a group of blue boxes on the diagram within the genes by the control ማለት ነው ሌላኛው ኖርማል ጂኖች ሌሎቹ በየሎ ወይም በቢጫ የተመለከቱት ናቸው and at least one class 3 gene when we see the 7s cell RNA gene is there it contains a weak internal promoter as well as a sequence in the 5 prime flanking region of the gene that is required for high level transcription 5 prime ላይ inside in the high level transcription የሚያስፈልግ box of gene ይኖራል and other class regions ለምሳሌ 7sk and u6 RNA genes የምንላቸው አሉ ቅድም ያየናቸው እዛ ጋር they lack internal promoters altogether and also they contain promoters that strongly resemble class 2 promoters to that reason they lie in the 5 prime flanking region and they contain the data box ቅድም ከፕሮሞተር 2 regions ጋር ይመሳሰላሉ ያልናቸው እዚህ ጋር the RNA polymerase 2 regions ያልናቸው -25 area ላይ data boxን ይዟል እነዚህ በዛ መልኩ ይመሳሰላሉ other class regions የምንላቸው ለ7sk እና ለ6 RNA genes የሚካተቱት ለዚህ ይሄ ፕሮፐርቲ ይኖራቸዋል ኢንተርናል ፕሮሞተሮች ባይኖራቸውም they have promoters that strongly resemble the class 2 genes that contain 5 prime flanking region and as well as the data box regions that make a similarity with the class 2 classical gene regions so the promoters and transcription factors for RNA polymerase 2 there are many genes in the transcribed by RNA polymerase 2 they have the data box ይኖራል or hogness box ይኖራል ከዛ ውጭ promoter consensus sequence is found about 25 nucleotides in the upstream ምሳሌ እዚህ ጋር class 1 regional from the center of transcription ከሁሉ ከዛ ወደ ኋላ ነው የሚመጣው so around the -25 region the data box which is found data box in eukaryotes you can also call it as the hogness box እንለዋለን it's a promoter region one of the promoter region and the CAAT box the second consensus region it is mostly found between the -70 and -80 regions of the nucleotides from the starting point of transcription ወደኋላ ስናየው ብዙውን ጊዜ GG ጓኒና ሳይቲዲን ሪች የሆኑ ኑክሊዮታይድ ቤዞች ይኖሩበታል እዚህ ሲኩዌንስ ላይ GG CC AT CT ይሆናል ሌላም ጊዜ ሊቀያየር ይችላል so the other genes for example those that are always or constitutively expressed are there and no data box is typically present in those genes instead in those genes as I told you before the GC rich region is found here region የት ነው -70 region ላይ ነው ብዙ ጊዜ ቀደም እንደነገርኳችሁ ወይም CAAT box ይባላል CAAT box ይባላል ግን CAAT box region ላይ GC regionዎች ወይም GC ኒውክሊዮታይዶች ብዙ ጊዜ ይበዛሉ። And because those sequences are on the same molecule of DNA as the gene being transcribed, and they are called the cis-acting elements rather than the promoter genes. ሁለቱም cis-acting elements ልንላቸው እንችላለን። So such sequences they serve as binding site for proteins, and those proteins are called the transcription factors, which can be used to facilitate the transcription process, and which in turn interact with each other and with RNA polymerase. RNA polymerase 2, sorry, because there are gene regions for RNA polymerase 2. In addition to binding of DNA, specific transcription factors also bind other proteins. Those other proteins, we can call them the co-activators, and recruiting those proteins to the transcription complex is important. For instance, the formation of closed complex begins when the TATA binding protein, shortly we can call it that, TBP, binds to the TATA box of the promoter region. Because in order to bind this the RNA polymerase with this region, TATA box is very important. ኋላ ዲያግራሙ ላይ ከስር ነው ታታ ቦክስ ናችን ኤሪያ ሄዶ አይደንቲፋይ ለማድረግ የሚያገለግል ታታ ቦክስ ታታ ባይንዲንግ ፕሮቲን TBP ነው የምንለው can be used to identify that promoter region of the TATA. ሌላኛው transcription factor 2A, its binding is there in the complex transcription complex, but although not always essential, it can stabilize transcription factor 2B na TATA binding protein complex on the DNA. ምክንያቱም እንዳልኳችሁ እያንዳንዶቹ እነዚህ transcription factor they do have their own function. ያልነው TATA binding protein can be used to identify the TATA box region and to bind with it ብለናል. And the TATA factor 2A ደግሞ it can be used to stabilize the TATA factor, the transcription factor 2B na TATA binding protein complex on the DNA, and can be important at non-consensus promoters where TATA binding protein binds is relatively weak. Then transcription factor 2F, it helps target polymerase 2 to its promoter region. Now, እነዚህ የተያያዙ ናቸው እያንዳንዳቸው የራሳቸው ፋንክሽን ስላላቸው. And this transcription factor 2F is interacting with the transcription factor 2B, and by reducing the binding of polymerase to non-specific site on the DNA. ምን ያህል የተያያዙ ናቸው የሚለውን ቅድም ለምሳሌ TATA binding protein አለ ብለን TATA binding protein ሲኖር transcription factor 2A አለ። ለምን ለመርዳት ነው የሚያገለግለው? transcription factor 2B TATA binding protein ጋር ኮምፕሌክስ እንዲፈጠር ለማድረግ ነው የሚረዳው, which is transcription factor 2A. Then ደግሞ transcription factor 2F ደግሞ it can help us to target RNA polymerase 2 ወደ ትክክለኛ promoter region ወይም transcription factor 2B እና TATA binding protein complex, even transcription factor 2A complex ወዳሉበት ቦታ ላይ ሄዶ directly በTATA binding protein መሪነት bind እንዲያደርግ ያገለግላል. So those are the transcription factors. Finally, the transcription factor 2E and transcription factor 2H binds to create the closed complex. አሁን ለምን እያንዳንዳቸው አክቲቪቲ ስላላቸው transcription factor 2H አለ. This transcription factor 2H has DNA helicase activity. It can be used to unwind the parental DNA strand during the process of transcription. ምክንያቱም unwinding ያስፈልገዋል. And that promotes the unwinding of DNA near the RNA transcription start site. And this process, as it is helicase activity, as we know that it requires ATP hydrolysis, and this transcription factor 2F is undergoing this ATP hydrolysis and by creating an open complex. Counting all the subunits of the virus essential factors, excluding transcription factor 2A. Now, its minimal active assembly has more than 30 repeptide chains. So, the transcription factor 2H ምንድነው activity ይባል? It has DNA helicase activity in the complex. It can be used to unwind the DNA strand. እያንዳንዶቹን እንዳየን ስለዚህ the proteins required for initiation of transcription process as the RNA polymerase II promoters of prokaryotes have their own functions. ለምሳሌ transcription process protein initiation ላይ polymerase II ሰብዩኒቶች አሉት ፋንክሽን ከ10,000 እስከ 220,000 ሰብዩኒቶች አሉት ፋንክሽኑም ካታላይዝ RNA ሲንተሲስ DNA polymerase II ካታ ባይንዲንግ ፕሮቲን ይዘ ቅድም እንዳልኳችሁ ትራንስክሪፕሽን ኮምፕሌክስ ፎርሜሽን ላይ አክቲንግ as facilitators ሆኖ ነው የሚያገለግለው it can be used to specifically recognize the tata box from the promoter region of the DNA parental DNA strand transcription factor 2A the subunit it has different subunit structures ነው ያለው composed of a lot of units ነው ለምሳሌ 12,000 19,035 ዩኒቶች የተለያዩ 3 ዩኒቶች ስለሆኑ ነው እሺ እዚህ ጋር 12 ዩኒቶች ሲባል ለምሳሌ ከ10,000 እስከ 220,000 የሚደርሱ እያንዳንዱ ላይ ያለ አንድ ዩኒት ነው 38,000 ሰብ ዩኒት እዚህ ጋር ደግሞ transcription factor 3A it has 3 subunits and one of the subunit has 12,000 molecular unit ነው the 19,000 the other one has 35,000 the major function ላይ ነው major transcription factor 2A stabilizes binding of the transcription factor 2B and theta binding protein to the promoter region bind ካደረጉ በኋላ እነሱን stabilize ለማድረግ ያገለግላል transcription factor 2B ደግሞ አንድ ሰብዩኒት ያለው ነው 35,000 molecular unit አለው it binds to theta binding protein and recruits polymerase II transcription factor 2F complex ነው እነዚህ ኮምፕሌክሶች መጥተው ከታታ ባይንዲንግ ፕሮቲን ጋር ባይንድ አድርገው ታታ ባይንዲንግ ፕሮቲኑ ያንን ኮምፕሌክስ መርቶ ወደ ፓረንታል ዲኤንኤ ስትራንዱ ታታ ባይንዲንግ ፕሮቲን ኤሪያ ላይ ታታ ባይንዲንግ ወይም ታታ ኤሪያ ላይ ታታ ቦክስ ፕሮሞተር ሪጅን ኤሪያ ላይ ባይንድ እንዲያደርጋቸው ነው transcription factor 2E which has two subunits 34,000 እና 57,000 molecular weight ያላቸው subunits ላይ ነው it recruits transcription factor 2H as I told you this transcription factor 2H has ATPase and it can undergo the helicase activity to unwind the parental DNA strand in the transcription complex transcription factor 2F ደግሞ ሁለት ሰብዩኒቶች አሉት number of subunits ሰብዩኒቶቹ 30,000 እና 74,000 ናቸው ብለናል it binds tightly to polymerase II binds to transcription factor 2B and prevents binding of polymerase II to non-specific DNA sequences ለምንድን ነው ከትራንስክሪፕሽን ፋክተር 2B ጋር ይሄኛው polymerase II ከትራንስክሪፕሽን ፋክተር 2B ጋር ባይንድ እንዲያደርግ ነው የሚያደርገው polymerase II ደግሞ በቀጥታ ሄዶ ትራንስክሪፕሽን ፋክተር 2B ጋር ባይንድ ሆነ ማለት ቅድም ትራንስክሪፕሽን ፋክተር 2B ፋንክሽኑ ምንድነው ብለናል it binds to theta binding protein and it recruits polymerase II እና transcription factor 2F complex for leading them with theta binding protein for leading them towards the theta site now okay the transcription factor 2 can form a complex with polymerase II and its function is to lead this the complex of polymerase II and this transcription factor F itself towards the theta binding protein area towards the theta binding protein area then the transcription factor 2H is there it has 12 subunits and those subunits have molecular weight starting from 35,000 to 80,000 molecular units as I told you before it has ATPase activity and helicase activity it's majorly used to unwind the parental DNA strand in order to in order to make or prepare free promoter regions identified by the polymerase and other protein complex activities. ስለዚህ እንደዚህ ነው ስታዩት diagrammatically we can express like this one. For instance, the center is there. DNA አለን. First data binding protein or transcription factor 2B and or transcription factor 2A complexes together can be binded with it. Then transcription factor 2B will bind. The transcription factor 2F and polymerase RNA polymerase 2 complexes are binding. ከዛ በኋላ ባይንድ ያደርጋሉ። transcription factor 2E bind ያደርጋል ቀጥሎ ከዛ transcription factor 2H bind ያደርጋሉ። ከዛ እነዚህ እንዳለ ወዴት ነው ታታ ባይንዲንግ ፕሮቲኑ ስላለ ቀጥታ ወደዚህኛው ቦክስ ይኖራቸዋል ባይንድ ያደርጋሉ። ባይንድ ካደረጉ በኋላ you can see here on this second diagrammatic structure the closed complex of the transcription can be formed. Then DNA unwinding to produce open complex ማለት ነው። ምክንያቱም እነዚህ ሁሉም ኮምፕሌክሶች ባይንድ ሆኑ ማለት transcription factor 2H can undergo the unwinding of the parental DNA as it has helicase activity. If the unwinding can take place means the phosphorylation of polymerase 2 can take place, initiation in promoter escape. The elongation process can be taking place ማለት ነው። ሌሎቹ ፕሮቲን ፋክተሮችም አሉ። ኢንዛይሙም ከዛ በኋላ ከፕሮቲን ፋክተሮች ተላቆ it starts to undergo the elongation process ማለት ነው። elongation process ከelongation process በኋላ እንግዲህ elongation process ሲቀጥል የሚያስፈልገው ፕሮቲን ፋክተርስ አሉ። elongation protein factors initiation ላይ የነበሩት ጨምሮ ለምሳሌ transcription factor 2H transcription factor 2E መልቀቅ ሲጀምሩ ወደ termination እየሄድን እንመጣለን። ኮምፕሌክሱን ለቀው ይወጣሉ። then elongation factors are there. እነዚህ elongation ፋክተሮችን ይወጡ ይመጣሉ። finally elongation factors and other those protein factors are leading to the complex ማለት it causes the termination of the newly synthesized RNA as we discussed previously, row dependent or row independent. Then polymerase RNA polymerase to release and the phosphorylation of the enzyme can be also taking place. On tata binding protein it should lead the enzyme to the tata box or the promoter box. This is the heart of the initiation complex. Now, the function of those all proteins is to identify the promoter region as well as to start and initiate the transcription process from the center of transcription. So the surface of the tata binding protein saddle provides docking site for the binding of other components. And additional transcription factors they can assemble on this nucleus in a defined sequence. And as we mentioned, the transcription factor 2A is recruited, followed by transcription factor 2B, and the transcription factor 2F and ATP-dependent helicases that initially separates the DNA duplex for polymerase activity to make it ready. Finally, RNA polymerase 2 and then transcription factor 2 join other factors to form a complex called the basal transcription apparatus. So you can see the transcription initiation in the others. Is that better to stop here? Hello?
አቤት
እዚህ ጋር ብናቆም ይሻላል ወይስ አርፈን እንቀጥል? እንዴት ነው?
5 ተኩል ሆኗል እስከ 6 ነው አይደል? ከዛ በኋላ ምንድነው የሚሄዱ ልጆች አሉ ክሊኒካል ቪዚት ዛሬና አሁን 5 ተኩል ነው እስከ 6 ነው አይደል ክላሱ?
እስከ 6።
ኦኬ እና ነገ ብንቀጥል ይሻላል ይቀራው?
እንዴት ይሻላል? ብሬክ ስላላረግን እንዴት ነው?
4 ተኩል አካባቢ ነበር ብሬክ መውሰድ የነበረብን።
ነገ እንቀጥል።
እሺ ቲቸር
ነገ ቀጥለን ከነገ ወዲያ አይ ቲንክ እንጨርሳለን። አርብ አይደል?
አርብ ነው።
ይበቃናል ያስባል።
እሺ ቲቸር ቴንክ ዩ።
ለመጨረስ ማለት ነው። ምክንያቱም እቺን ጨርሰን ከዛ ትራንስሌሽን ነው የሚቀረው። እዚህ ላይ ወደ 20 ስላይድ አካባቢ ነው
18 ይቀራል። እሺ
ስለዚህ have a nice time እንግዲህ ሪከርዲንጉ የባለፈውን አያችሁ ጥራት አለው? ድምፁ ይሰማችኋል?
ይሰማ አንድ አይነት ነው። እንደዚህ አይነት ነው
እኮ ጥራት አለው። ይሰማል?
አዎ ይሰማል ይሰማል።
ኦኬ ስለዚህ ኖቱን ልክላችኋለሁ። ይሄን
ኖቱንም ሪከርድ
አድርጓል።
እሺ እና አንድ ጥያቄ አለ ቲቸር።
ኦኬ ጥያቄ
ኢንኮል ሜሴጅ ውስጥ አለ።
Okay, the difference between basal and optimal transcription. ብዙ ጊዜ ኦፕቲማል ትራንስክሪፕሽን ላይ እዛ ውስጥ ያሉት ጂኖች ስፔሲፊካሊ the synthesized RNA molecules will not undergo much more type of post transcriptional processes modifications ነው። ብዙ ፖስት ትራንስክሪፕሽናል ሞዲፊኬሽን ላይ ያስፈልጋቸው ይችላል። ለምሳሌ በኦፕቲማል ትራንስክራይብድ የሚደረጉት ሜቢ ትራንስፈር RNA ሊሆን ይችላል፣ ራይቦዞማል RNA ሊሆን ይችላል which do not require much more excess post transcriptional modifications። ባዛል ላይ ግን ሲሆን ሞስትሊ በባዛል ሲንተሳይድ የሚደረገው ሜሴንጀር RNA ሊሆን ይችላል and it is required to undergo the post transcriptional process splicing splicing 3 prime tailing and 5 prime cutting በደንብ የሚያከናውኑ ይሆናል ማለት ነው። Have you got my point? Have you got my point? Okay. Okay. Do you have another question? Do you have another? ለጥያቄ የላችሁም? Okay. ለጥያቄ ከሌላችሁ have a nice time and I will continue tomorrow morning.
Okay, thank you.